Welcome and good evening. Tonight, as the room grows quiet and the world beyond you begins to soften into darkness, there is something comforting to remember. You are never truly alone. Even now, while you rest beneath a blanket, while your breathing becomes slower, while your thoughts begin to loosen their grip on the day, an ancient and astonishing system is quietly caring for you. It has no single headquarters. No central commander sits somewhere inside your body giving orders. Instead, protection is spread through your blood, your skin, your lungs, your digestive system, your lymph nodes, your bone marrow, and countless microscopic spaces between your cells. Together, these defenses form your immune system, and for most of your life, they work so quietly that you barely notice them. Every breath brings the outside world into intimate contact with you. Every meal carries thousands of unfamiliar molecules through your digestive tract. Every object you touch leaves traces of the environment upon your skin. And yet, through all of this contact with a living world, the body maintains a remarkable sense of order. It knows, most of the time, what belongs. It recognizes what should be tolerated. And when something potentially harmful appears, it has ways of responding. Some defenses act within minutes. Others take hours or days. Some remember encounters that happened years before. And some are so old, in evolutionary terms, that versions of them existed long before human beings walked beneath the stars. So tonight, we are going to take a slow journey through this hidden world. There will be no battles to worry about here. No need to imagine your body as a place of constant warfare. Instead, think of it as a peaceful country at night. A country with walls, rivers, roads, watchtowers, libraries, gardens, and quiet patrols. Most nights are uneventful. Most visitors are harmless. Most of the microscopic creatures that share our world never cause us trouble at all. And through it all, your body keeps watch with extraordinary patience. So settle a little more deeply into wherever you are resting. Allow your shoulders to become heavy. Let your jaw relax. There is nowhere else you need to be. For the next little while, we are simply going to wander through the quiet landscape within you, beginning at the very edge of your body, with something so familiar that we rarely stop to appreciate it. Your skin. Imagine, for a moment, that you could become impossibly small, smaller than a grain of sand, smaller than a speck of dust drifting through a beam of afternoon sunlight. You descend slowly toward the surface of your own hand. From an ordinary distance, skin looks smooth, but as you approach this landscape at microscopic scale, it changes. Fine ridges rise around you. Tiny openings appear. Delicate hairs stand like narrow trees. There are valleys and folds and patches of moisture. And across this enormous terrain lives an entire community of microorganisms. Many are harmless. Some are useful. They have lived with human beings for longer than recorded history. Your skin, then, is not simply a wrapper around the body. It is a living boundary. The outermost portion of the skin contains layers of tightly arranged cells, including cells that have become rich in a strong protein called keratin. These outer layers create a remarkably effective physical barrier. For a microorganism outside your body, healthy intact skin is not an easy landscape to cross. It is dry in many places. It is slightly acidic. It contains oils and other substances that can make life difficult for certain unwanted microbes. and it is constantly renewing itself. Cells form deeper in the epidermis gradually move outward. Over time, they reach the surface. Eventually, they are shed, quietly, continuously. You have been leaving tiny pieces of yourself behind throughout your entire life. On bed sheets, on clothing, in rooms, in the dust of old houses. And although this sounds almost poetic, it is also practical. The surface of your body is always being renewed. Your first great defense is not a dramatic response. It is simply a wall that is always rebuilding itself. Now imagine walking across this microscopic landscape as evening settles. The surface is warm. There is a faint sheen of natural oil. Around you are bacteria that normally live peacefully upon human skin. The word bacteria sometimes makes people uneasy, but the overwhelming story of microbes is far more interesting than a simple division between good and bad. The world is filled with microorganisms. They live in oceans. They live in soil. They float through the atmosphere. They inhabit deep underground environments and icy landscapes. And they live upon us. Many of the microbes that normally occupy our skin simply go about their microscopic lives without causing disease. Their presence can even make it harder for less welcome organisms to establish themselves. Space is already occupied. Resources are already being used. The neighborhood, in a sense, already has residents. This idea will return later when we visit the digestive system, where the relationship between human beings and microbes becomes even more extraordinary. For now, though, we continue across the skin. Ahead is a tiny break in the surface. Perhaps it came from a scratch so small you never noticed it. Suddenly, the boundary is no longer perfect, and beneath that opening lies another world. Here, the immune system has prepared for exactly this possibility. Cells living in tissues can detect signs of damage. Certain immune cells are already present nearby. Chemical signals begin to change the local environment. Blood vessels may widen. Their walls can become more permeable, allowing fluid and immune cells to move more easily into the affected tissue. From the outside, you might notice redness, perhaps warmth, perhaps slight swelling. We call this inflammation. The word can sound unpleasant because excessive or prolonged inflammation can certainly become harmful. But in its ordinary, controlled form, inflammation is one of the body's oldest protective responses. It helps bring resources where they are needed. It is less like an alarm bell ringing through the entire kingdom, and more like a lantern being lit beside a particular road. Something happened here. Come and take a look. Among the cells that may respond are remarkable wanderers known as neutrophils. Neutrophils are a kind of white blood cell. They are produced in enormous numbers in the bone marrow, and they spend part of their lives circulating through the bloodstream. When signals indicate that certain tissues need help, neutrophils can leave the blood and move toward the affected area. Imagine them not as soldiers, but as night workers. The kind of people who arrive after a storm has passed through a town. They inspect, they contain, they clear debris, they help restore order. Neutrophils can engulf certain microbes in a process called phagocytosis. The word comes from Greek roots associated with eating. And under a microscope, the description makes sense. The cell extends its membrane around a particle or microorganism. Slowly, the target is enclosed. It is brought inside. Then specialized cellular compartments and chemical processes help break it down. Another group of immune cells, macrophages, are also skilled at this kind of engulfing and cleanup. Macrophages live in many tissues throughout the body. Their name roughly suggests big eaters, but their work is more sophisticated than the name implies. They can remove microbes. They clear damaged cells and cellular debris. They release signals that influence other immune cells. And they help connect the body's immediate defenses with more specialized immune responses. In different tissues, related cells develop distinctive characteristics. The lungs have their own resident macrophages. The liver contains specialized macrophages called Kupffer cells. The brain has immune-related cells called microglia, which perform important surveillance and maintenance within the nervous system. The body has adapted its guardians to the landscapes in which they live, just as a lighthouse keeper works differently from a forest ranger. The cells protecting one tissue may have responsibilities shaped by that particular environment. We leave the skin now and drift inward. Not far, just beneath the surface. Here lies a network of tiny blood vessels. Some are so narrow that red blood cells pass through almost one at a time. Imagine entering one of them. The walls surround us like a soft, living tunnel, and suddenly we are carried forward by the gentle current of blood. The bloodstream is one of the great transportation systems of the human body. Red blood cells dominate the view. There are trillions of them in the body, endlessly carrying oxygen from the lungs toward tissues and helping transport carbon dioxide in the opposite direction. They are flexible little discs, shaped in a way that gives them a large surface area. Around them drift platelets, and scattered among this vast red population are white blood cells. Immune cells. They are much less numerous than red blood cells, but their variety is extraordinary. Neutrophils, lymphocytes, monocytes, eosinophils, basophils. Each belongs to a slightly different story. Each has its own tendencies, abilities, and role. And all of them began, in one way or another, from an even deeper source. To understand where these cells come from, we must travel into the bones. Imagine drifting toward the center of a large bone. The hard outer structure gives way to a softer interior environment. Here is bone marrow. At first, the name may suggest something simple: a filling, a soft substance inside a hard shell. But biologically, bone marrow is one of the busiest places in your body. It is a nursery of blood. Within red bone marrow are hematopoietic stem cells. These remarkable cells give rise to the major cellular components of blood. From branching developmental pathways emerge red blood cells, platelets, and many kinds of white blood cells. All day, all night, whether you are awake or sleeping, the production continues. Some blood cells live for months. Some immune cells may be relatively short-lived. Others can remain for years. So the marrow must constantly adjust and replenish the population. Imagine a quiet underground workshop that never closes. There is no sound of machinery, no hammering, no footsteps, only chemistry. Signals bind to receptors. Genes become active or quiet. Cells divide. Young cells mature. And eventually, when they are ready, many enter the circulation. You have carried this hidden nursery inside your skeleton since childhood. Even now, while you lie perfectly still, new blood cells are being formed within you. It is difficult to imagine a more peaceful kind of industry. Among the cells beginning their lives here are lymphocytes. Two groups will become especially important to our story: B cells and T cells. B cells mature in the bone marrow. T cells begin from precursors in the marrow too, but their development leads them to another remarkable place: the thymus. So we leave the marrow and drift upward, past the blood, past the soft movement of the lungs, toward the upper chest, behind the breastbone. Here rests the thymus. In children, the thymus is relatively prominent and active. After puberty, it gradually becomes smaller and much of its tissue is replaced by fat, though it retains immune functions. Its importance early in life is enormous. The thymus is where developing T cells undergo an extraordinary education. And perhaps education is one of the best ways to imagine it. A young T cell cannot simply be released into the body with the ability to react strongly to whatever it encounters. That would be dangerous. The immune system faces a profound challenge. It must respond to potential threats while avoiding destructive attacks against the body's own healthy tissues. So developing T cells are tested. Through complex biological selection, cells that cannot function appropriately are removed. Cells that react too strongly against the body's own molecules are also largely eliminated. Only a fraction complete the process successfully. It is one of the quiet miracles of immune development. Before many T cells are permitted to wander through your body, they must first learn something very close to restraint. Protection depends not only on knowing when to respond, it depends on knowing when not to. This balance is at the heart of immunity. Your body is not trying to destroy everything unfamiliar. If it did, ordinary life would become impossible. Every meal introduces molecules from other organisms. Every breath contains particles from the environment. Your body hosts enormous communities of microbes. Pregnancy presents the immune system with another remarkable biological challenge, requiring finely regulated interactions between maternal tissues and a developing fetus. Life requires tolerance. So the immune system is not merely a defense system. It is a system of judgment, recognition, communication, memory, and restraint. Imagine a wise old city whose gates are busy from sunrise until long after dark. Merchants arrive, travelers come and go, animals move through the streets, letters are delivered, food enters the marketplace. Most of this activity is ordinary. The city does not close its gates every time someone unfamiliar appears. Instead, it watches. It learns patterns. It notices disturbances. And only when certain signs suggest trouble does the response begin to change. Your immune system works in a similarly nuanced world. Now we travel upward again, toward the nose, toward the mouth, toward the passages where the outside world enters you most frequently. Every day, thousands of liters of air may move through the respiratory system, depending on activity and breathing rate. The air carries oxygen, of course, but it also carries dust, pollen, tiny droplets, microorganisms, fragments of plants, particles too small to see. Your lungs are delicate. The surfaces where oxygen passes into the blood must be thin. They cannot simply be covered in something resembling thick armor. So the respiratory tract uses other forms of protection. Inside much of the airway is a moist lining. Mucus traps particles. And along sections of the respiratory tract are microscopic hair-like structures called cilia. Cilia move in coordinated waves. Their motion helps carry mucus and trapped material upward, away from the deeper lungs. Imagine standing beside an enormous field of tall grass at night. A breeze passes over it. One row bends, then another, then another. A smooth wave travels across the landscape. Cilia are far smaller than blades of grass, of course, but their coordinated movement can create something equally graceful. Wave after wave, minute after minute, helping move unwanted material toward places where it can be swallowed or expelled. You rarely notice this work. That is the strange beauty of the body's defenses. Their greatest successes often feel like nothing at all. A cough can also protect the respiratory tract. So can a sneeze. They are sudden, powerful ways of moving irritating material. But on this peaceful journey, we need not linger on their force. Instead, follow the mucus slowly toward the throat. Some of it is swallowed. And now we arrive at another entrance to the body, the digestive system. This is one of the busiest borders anywhere within you. Food carries the chemistry of the outside world directly into the body. And yet digestion requires openness. Nutrients must eventually cross from the intestine into the internal environment. Water must be absorbed. Minerals and other molecules must pass through. So once again, the body needs a boundary that can distinguish between ordinary material and signs of danger. The stomach provides one kind of defense. Its acidic environment helps destroy many microorganisms that arrive with food. Not all of them, certainly, but the stomach is far from an easy place to survive. Then, farther along, we enter the intestines. And here, the landscape becomes astonishing. The human gut is home to a vast community of microorganisms. Bacteria are especially abundant, along with other microbes. Collectively, these communities are often called the gut microbiota. Their combined genetic material and broader ecological functions are sometimes discussed as the microbiome, though the terms are used in slightly different ways. Whatever name we choose, the central idea is beautiful. You are an ecosystem. Human cells do not live in isolation. Your body developed in constant relationship with microscopic life. Gut microbes can help process components of food. They produce various compounds. They interact with intestinal cells, and they influence the development and regulation of the immune system. The relationship is complicated. It varies from person to person. It changes with diet, age, medication, environment, and many other factors. Scientists are still discovering just how deep these relationships go, but we already know enough to appreciate the quiet wonder of it. Imagine the intestine not as a simple tube, but as a long, warm river valley. Along its banks are countless inhabitants. Some live close to the protective mucus layer. Others occupy different ecological niches. Nutrients flow through. Chemical messages pass between species. Human cells sense what is happening nearby. Immune cells wait beneath the surface. It is crowded beyond imagination. And yet, most of the time, there is peace. The lining of the intestine is only one cell layer thick in many places. That sounds fragile, but these cells are tightly connected. They create a selective barrier between the contents of the gut and the tissues beyond. Mucus adds another protective layer. Antimicrobial molecules contribute further defenses. And one particular antibody, called immunoglobulin A or IgA, is especially important along mucosal surfaces. Antibodies are proteins made by specialized descendants of B cells. We will meet them properly later. For now, imagine IgA molecules quietly patrolling the moist boundaries of the body, the intestines, the respiratory tract, other mucosal surfaces. They can bind to particular targets and help prevent certain microbes from attaching to or crossing epithelial surfaces. Again, the strategy is elegant. The body does not always need to destroy something. Sometimes it is enough simply to prevent it from going farther. A locked door can be more peaceful than a confrontation. Scattered throughout the intestinal immune system are organized tissues that sample and monitor the environment. In the small intestine, structures called Peyer's patches are part of this network. Immune cells here encounter information about what is passing through the gut. The challenge is immense. Food is foreign. Helpful microbes are foreign. Harmless environmental molecules are foreign. Yet most should not trigger destructive immune responses. So the intestinal immune system spends its life performing one of biology's most delicate balancing acts. Watch carefully. Respond when necessary. Remain tolerant when appropriate. Maintain the barrier. Communicate with neighboring cells. And then, return to quiet. There is a lesson in this, perhaps. Strength is not constant action. Sometimes strength is the ability to remain calm in the presence of something unfamiliar. From the intestine, we follow a tiny vessel into another network. Not a blood vessel this time, a lymphatic vessel. The lymphatic system is easy to overlook because it does not have a single powerful pump like the heart, yet it reaches throughout much of the body. Fluid naturally leaves blood capillaries and enters tissues. Much of it eventually returns to circulation through the lymphatic system. Once this fluid enters lymphatic vessels, we call it lymph. Imagine a network of quiet streams running beside the faster rivers of the bloodstream. These streams collect fluid from tissues. They carry proteins. They carry cellular debris. They may carry immune cells and material from microbes. And along these streams are small structures called lymph nodes. You may have felt them before, perhaps beneath your jaw when you were ill. Perhaps in your neck. Lymph nodes are found in many regions, including the neck, armpits, chest, abdomen, and groin. Usually they are small and unnoticed. But inside them, extraordinary conversations take place. Let us enter one. The current slows. The node surrounds us like a quiet station beside a river. Lymph arriving from nearby tissues brings information, fragments of proteins, signals of what has been happening upstream. Immune cells move through organized regions. Among them are B cells and T cells. And here we meet another important character, the dendritic cell. Dendritic cells are exceptionally skilled at gathering material in tissues and presenting pieces of it to T cells. Their name comes from their branching appearance, which reminded scientists of trees. Imagine one now, a small living tree moving through microscopic darkness. Its branches extend outward. It samples its surroundings. If it encounters certain signs associated with infection or tissue disturbance, its behavior can change. Some dendritic cells travel toward lymph nodes, and there, they display molecular fragments on their surfaces. These displayed fragments are called antigens when they are recognized by immune receptors. A T cell passing through the node may inspect what the dendritic cell is presenting. Most T cells will move on. Their receptors do not match. But somewhere among the enormous diversity of T cells, there may be one whose receptor recognizes that particular molecular shape. And when the appropriate additional signals are present, something remarkable begins. To appreciate this moment, we need to understand just how diverse your adaptive immune system is. Each developing B cell and T cell produces a distinctive receptor through a fascinating process of genetic rearrangement. Segments of DNA are recombined during development. The result is an enormous variety of possible receptors. You do not need a separate gene for every pathogen you might encounter. Instead, your immune system generates a vast library of receptors in advance. A library filled with possibilities. Imagine entering an ancient circular room. The ceiling disappears into darkness. Shelves rise around you. Millions upon millions of books line the walls. Most will never be opened. Some may wait your entire lifetime without ever finding the thing they were designed to recognize. But the library is ready. When a new molecular pattern appears, the immune system searches not by consciously designing a new receptor from nothing, but largely by finding lymphocytes whose receptors already fit. The right book was waiting on the shelf. Suppose a T cell recognizes the antigen presented by a dendritic cell. Recognition alone is not always enough. Immune activation is carefully regulated. Additional molecular signals help determine whether the T cell should truly respond. When the conditions are appropriate, the T cell begins to change. It activates. It divides. One cell becomes two. Two become four. Four become more. A small population of cells sharing the same useful receptor begins to expand. This is called clonal expansion. The body is making copies of something that has proven useful. Some T cells can help coordinate immune responses. Certain helper T cells communicate with B cells and other immune cells through direct contact and signaling molecules. Other T cells, including cytotoxic T cells, can recognize body cells displaying certain abnormal or foreign-derived antigens and can induce those cells to die. This becomes particularly important when viruses enter cells, because viruses present the immune system with a special problem. A bacterium may exist outside your cells. A virus cannot reproduce that way. Viruses carry genetic material enclosed within structures made of proteins and, for many viruses, membranes. To make more copies of themselves, they must enter living cells and use cellular machinery. So imagine a virus reaching one of your cells. It enters. Its genetic instructions begin influencing the cell. New viral components are produced. From the outside, the infected cell may still resemble its neighbors. But cells have a way of displaying tiny samples of proteins from inside themselves on molecules at their surface. These molecules include a group called the major histocompatibility complex. In humans, they are often discussed as HLA molecules. You can imagine them as little display windows. Small protein fragments are brought to the surface. Passing T cells can inspect these displays. A cytotoxic T cell with the appropriate receptor may recognize that something is wrong. If the proper conditions are met, It can trigger the infected cell to undergo a controlled form of death. The infected cell is sacrificed to protect the larger community. Quietly, locally, precisely. But long before specialized T cells arrive, another group may already be watching. Natural killer cells. Their name sounds dramatic, but their role is carefully regulated. Natural killer cells belong to the body's innate immune defenses. Unlike T cells, they do not depend on the same kind of highly specific antigen receptor created through gene rearrangement. Instead, they integrate signals from cells around them. Healthy cells display molecular signs that help communicate normality. Some infected or transformed cells alter these signals. Natural killer cells can detect certain suspicious patterns and may cause abnormal cells to die. Imagine them as inspectors walking through a sleeping town. They do not carry a photograph of one particular intruder. Instead, they notice when a house no longer looks quite right. A window that should be lit has gone dark. A familiar signal is missing. Another unusual signal has appeared. They pause. They investigate. And when the balance of information indicates danger, they act. The immune system uses many different styles of recognition because no single strategy would be enough. Now return with me to the lymph node. The B cells are waiting. Each carries its own version of a receptor on its surface. This receptor is essentially a membrane-bound form of antibody. When a B cell encounters a matching antigen, and when it receives the appropriate additional signals, often including help from certain T cells, it can become activated. Then it divides. Some of its descendants become plasma cells. Plasma cells are antibody factories. They can produce extraordinary quantities of antibodies. These Y-shaped proteins are released into body fluids, and each antibody produced by a particular plasma cell lineage is shaped to recognize a particular molecular target. Imagine millions of tiny keys flowing into the bloodstream. That comparison is not perfect because antibodies do not unlock things. They bind. Perhaps it is better to imagine millions of carefully shaped tags. When the correct tag encounters its matching structure, it attaches. That attachment can change what happens next. Antibodies can block certain viruses or toxins from attaching to cells. They can coat microbes, making them easier for phagocytic cells to recognize and engulf. Some antibodies can help activate another ancient defense system called complement. And all of this begins with molecular recognition. A shape meeting a shape. Chemistry recognizing chemistry. There are several major classes of antibodies. IgM often appears prominently early in a first immune response. IgG is abundant in blood and tissues and plays many roles in longer-term protection. IgA, as we encountered earlier, is especially important at mucosal surfaces and is also found in secretions such as saliva and breast milk. IgA participates in defense against certain parasites and also plays a central role in allergic reactions. IgD is found mainly on the surfaces of certain B cells and contributes to their activation and regulation. You do not need to memorize any of these names tonight. Let them drift past. IgM, IgG, IgA, IgA, IgD. Just letters carried gently downstream. What matters is the deeper idea. The body has not invented one universal antibody. It has created a flexible system capable of generating extraordinary specificity. And once a useful response has been made, the immune system may remember. Memory is perhaps the most beautiful part of this entire story. After an infection has passed, most of the expanded immune cells are no longer needed. The population contracts. The body returns toward its ordinary state. But some cells remain. Memory B cells, memory T cells. Long-lived plasma cells may continue producing antibodies for extended periods, depending on the particular immune response. The exact duration of immune memory varies enormously. Some responses fade. Others can persist for many years. But the principle is remarkable. An encounter can leave behind information. The next time the same pathogen appears, the adaptive immune system may respond more quickly and effectively. Your body carries pieces of its history, not as conscious memories, not as images, not as words, but as cells. Think for a moment about childhood. Perhaps there was a house you once knew, a window where afternoon light fell across the floor, the sound of rain against a roof, the smell of wet earth after a storm. Maybe those details are still somewhere in your mind. Your immune system has memories too, different memories, older than many of the stories you consciously remember. A B cell lineage may carry information shaped by an encounter from years ago. A population of memory T cells may remain ready because of something your body experienced long before tonight. You have forgotten countless ordinary days of your life, but your immune system may still carry biological traces of some of what happened during them. It is a quiet archive, a library written in living cells. And this brings us naturally to one of medicine's most elegant ideas: vaccination. Long before scientists understood antibodies, lymphocytes, genes, or viruses in modern molecular detail, people noticed something important. Those who recovered from certain infections often seemed protected from getting the same disease again. Over centuries, different cultures experimented with ways of deliberately producing protection. One famous early practice was variolation against smallpox. It carried real risks because it involved material from smallpox itself. Then, near the end of the 18th century, the English physician Edward Jenner helped demonstrate a safer approach involving cowpox exposure to protect against smallpox. The word vaccine eventually grew from the Latin word associated with cows. But the science of vaccination would expand far beyond Jenner's early work. Modern vaccines can use several different approaches. Some contain weakened forms of pathogens. Some contain inactivated organisms. Some contain purified components. Some deliver genetic instructions that allow cells to briefly produce a harmless antigen so the immune system can learn to recognize it. The technologies differ. The underlying idea is wonderfully simple. Give the immune system information before the dangerous encounter happens. Let the library prepare the right books. Let memory form while the risk is controlled. Then, if the real pathogen arrives later, the body is no longer meeting it as a complete stranger. Smallpox provides one of humanity's most extraordinary examples. For centuries, smallpox caused immense suffering around the world. Then vaccination campaigns gradually pushed the virus back. Cases declined. Transmission chains were interrupted. Health workers searched for outbreaks and vaccinated people around them. Country after country eliminated the disease. The last naturally occurring case of variola major was recorded in 1975 and the last naturally occurring case of varicella minor occurred in Somalia in 1977. In 1980, the World Health Organization declared smallpox eradicated. A human disease that had haunted civilization for centuries was gone from natural circulation. And behind that achievement was immune memory. Millions of individual immune systems, each taught to recognize something before it arrived. It is difficult to imagine a quieter form of collective power. Now let us return to your body. The lymph node fades behind us. We enter the bloodstream again. The current carries us toward the upper left side of the abdomen. There, tucked beneath the ribs, lies another immune organ, the spleen. The spleen is soft, richly supplied with blood, and often overlooked until something goes wrong with it. But it performs several important tasks. It helps filter the blood. It removes aging or damaged red blood cells. And it provides spaces where immune cells can encounter materials circulating through the bloodstream. If lymph nodes are checkpoints along the lymphatic streams, the spleen is more like a great harbor on the blood river. Everything flowing through the circulation eventually participates in this enormous movement. Red cells, platelets, proteins, immune cells, and sometimes microorganisms or their components. Within the spleen, specialized immune regions examine what passes by. Once again, the body is sampling, observing, comparing, deciding. The spleen also reminds us that immunity is inseparable from the rest of physiology. The body is not divided into independent departments. The immune system talks to the nervous system. It interacts with hormones. It responds to signals from metabolism. It is influenced by sleep. It changes with age. It communicates with tissues throughout the body. Even the distinction between an immune cell and an ordinary tissue cell is not always as sharp as we might imagine. Many cells can detect danger signals. Many can produce signaling molecules. Many participate in local defense. The body behaves less like a machine assembled from separate pieces, and more like an orchestra. No instrument contains the music by itself. The music emerges from relationships. And now, as our journey grows deeper, we arrive at one of the oldest systems in your blood. Complement. Complement is not a single molecule. It is a collection of proteins that circulate mostly in inactive forms until particular pathways trigger them. Once activated, complement proteins can participate in a cascade. One activates another, then another. The signal grows. Certain complement fragments help promote inflammation. Others coat microbial surfaces and make them easier for phagocytes to recognize. And in some situations, complement components assemble into structures capable of damaging the membranes of certain target cells. The system is powerful, so naturally, it must also be controlled. Your own cells carry regulatory proteins that help prevent complement from damaging healthy tissue. Here again, we find the same principle: protection and restraint, action and control. The immune system survives by maintaining both. Imagine standing beside a mountain stream. A single drop falls from a leaf. It joins another. The water moves over stone. One small current meets the next. Soon there is enough water to turn a wheel. That is the feeling of a biological cascade. A small initiating event becomes amplified through a sequence of molecular steps. Blood clotting uses cascades too. Many cellular signaling systems do something similar. Biology often works this way because cascades allow tiny signals to become meaningful responses. But cascades also need breaks. Without regulation, amplification becomes dangerous. So the body builds inhibitors alongside activators, locks beside keys, brakes beside accelerators, silence beside sound. It is a pattern repeated everywhere inside you. By now, perhaps, the room around you feels farther away. Your breathing may have become slower without you trying. The body does many things best when we stop trying to supervise it. Your heart does not wait for instructions from your conscious mind. Your bone marrow does not ask permission to produce another neutrophil. Your lymph nodes do not wake you in the middle of the night to report their findings. They simply continue, quietly, patiently, and sleep itself is woven into this story. Sleep and immunity influence one another in complex ways. During sleep, patterns of hormones and signaling molecules change. Immune activity follows daily rhythms. Research has shown that adequate sleep supports healthy immune function, while prolonged sleep loss can disturb aspects of immune regulation. This does not mean that one perfect night of sleep creates an invincible immune system. Biology is never that simple, but sleep is part of the environment in which immune regulation occurs. So tonight, as you rest, you are not abandoning your body's defenses. You are giving your body something it has evolved to expect: a period of darkness, stillness, reduced activity, time. Long before electric lights, before cities glowed through the night, human beings lived beneath a powerful rhythm. Day arrived, light entered the eyes, activity increased. Then evening came, temperatures fell, shadows lengthened, and darkness returned. The immune system, like many other parts of physiology, is influenced by circadian rhythms. Certain immune cells change their patterns of movement across the day. Hormones associated with the sleep-wake cycle can influence immune activity. The body does not perform every task at the same intensity at every hour. It has timing, and perhaps that is comforting. You do not need to remain alert all night. Your body understands darkness. It has known darkness for millions of years. Let us drift now toward the lungs. The chest rises, then falls. A breath enters. Air travels through the nose or mouth, down the throat, through the trachea, and into branching airways. The branches divide again, and again, and again, until finally we arrive at tiny air sacs called alveoli. There are hundreds of millions of alveoli in healthy adult lungs. Their combined surface area is enormous. Here, oxygen crosses into the blood. Carbon dioxide moves in the opposite direction. The barrier must be extraordinarily thin, and because it is thin, it must be watched. Resident immune cells patrol the air spaces. Alveolar macrophages move across these delicate surfaces, engulfing particles and microorganisms that reach this far. Imagine them walking slowly across a shoreline at dawn. Behind them lies the immense ocean of air. Before them flows the dark river of blood. They stand between worlds. Most particles never reach the alveoli. The nose filters larger material. Mucus traps more. Cilia carry material upward. Coughing can clear the airways. Antibodies and antimicrobial substances add further protection, layer upon layer. This is another important principle of immunity: redundancy. The body rarely depends upon one perfect defense. Instead, it builds several imperfect defenses on top of one another. Skin, mucus, chemical barriers, resident microbes, phagocytes, complement, antibodies, T cells. Each has weaknesses. Together, they create resilience. A castle with only one wall may fall when the wall is breached. A living body uses walls, rivers, scouts, memory, repair crews, and diplomacy. And perhaps most importantly, it rebuilds. Repair is inseparable from defense. When tissue is injured, immune cells participate not only in clearing microbes, but also in coordinating healing. Macrophages can shift their behavior as conditions change. Early inflammatory signals may help contain damage. Later signals can support tissue repair and resolution. Fibroblasts produce extracellular matrix. New blood vessels may grow. Epithelial cells divide and migrate. Eventually, if all goes well, the disturbance fades. The tissue may return almost completely to its previous state, or a scar may remain. A scar is not failure. It is evidence that the body chose closure over perfection, a memory written in collagen. Think of an old tree. A branch once broke in a storm. Years passed. The wound closed. New wood formed around the damaged place. The tree never became exactly what it was before, but it continued growing. Human tissue often does something similar. Healing is not always restoration to an untouched past. Sometimes it is adaptation. The body repairs what it can. It reinforces what remains. Then life continues. We move now toward another quiet part of the immune system, the tonsils. At the back of the throat, lymphoid tissues occupy strategic positions near entrances used by air and food. They are part of a broader ring of immune tissue around the pharynx. Here, the body samples material arriving from the outside world. Children often have relatively prominent tonsils because the immune system is still encountering and learning about many antigens. Sometimes tonsils become inflamed. Sometimes they become enlarged. But on an ordinary night, they simply remain where they are, watching the crossroads. Nearby is saliva. It seems simple, mostly water. But saliva contains enzymes, proteins, antibodies, and antimicrobial substances. The mouth itself is home to a complex microbiota. Again, the goal is not sterility. A healthy human body is not sterile. Not on the skin, not in the mouth, not in the intestine. Life exists beside life. The immune system evolved within this crowded biological world. Its purpose is not to create emptiness. Its purpose is to preserve a workable relationship between self, environment, and microbial neighbors. This is a much gentler way to understand immunity, not endless war. Ecology. Now imagine the mouth at night. The room is silent. Your tongue rests. The jaw is loose. Saliva moves slowly across the tissues. Microbes continue their microscopic lives. Epithelial cells renew themselves. Immune molecules remain present. Blood flows through tiny vessels beneath the surface. Nothing has stopped because you are asleep. The scale of activity inside a resting human body is almost impossible to comprehend. Billions of cells are metabolically active. Proteins are being assembled. Old molecules are being broken down. DNA is being copied and dividing cells. Signals are traveling across membranes. And yet from the outside, You may appear perfectly still. That contrast is beautiful. Stillness outside, a universe within. Let us return to one of the immune system's greatest mysteries. How does it know what belongs to you? Every nucleated cell in your body carries molecular signatures. Proteins on cell surfaces help cells interact with one another and with the immune system. As we saw earlier, HLA molecules display fragments of proteins for inspection by T cells. The genes encoding these molecules are highly variable among people. This diversity is one reason tissue transplantation between unrelated individuals can be difficult. A transplanted organ may be recognized as foreign by the recipient's immune system. Modern transplantation medicine, therefore, depends on careful matching, immune-suppressing medications, and close monitoring. The same immune system that protects us from infection can also react strongly to transplanted tissue. There is no contradiction. The immune system is doing what evolution shaped it to do. It is responding to biological difference. Medicine has simply learned ways to negotiate with that response. Autoimmune diseases reveal another side of the same delicate balance. Normally, multiple mechanisms help prevent immune cells from attacking healthy self-tissues. But these mechanisms are not infallible. In autoimmune conditions, immune responses become directed against components of the body's own cells or tissues. Different autoimmune diseases affect different organs and biological pathways. Their causes can involve combinations of genetics, environment, immune regulation, and other factors that researchers continue to investigate. We do not need to dwell on illness tonight. The important point is simpler. Tolerance requires active work. The immune system is constantly solving a difficult problem. Protect the body without harming the body. Most of the time, it succeeds so completely that you never notice the problem existed. Allergies represent another kind of immune misjudgment. Pollen, for example, is usually harmless. It is simply plant material drifting through the air. Yet in someone with a pollen allergy, the immune system reacts to particular pollen proteins. IgA antibodies can bind to cells such as mast cells. When the relevant allergen appears again, it can trigger these cells to release histamine and other mediators. Blood vessels change. Mucus production increases. Nerves are stimulated. Eyes may water. The nose may run. A sneeze arrives. The immune system has mistaken something harmless for something requiring a strong response. Even here, though, we can see how carefully interconnected the system is. Antibodies, cells, chemical mediators, blood vessels, nerves, muscles, all participating in one response. Mast cells themselves are fascinating. They live in tissues, especially near boundaries with the outside world. The skin, the respiratory tract, the digestive tract. They contain granules filled with powerful chemical mediators. When activated appropriately, mast cells can contribute to defense and tissue responses. But in allergy, the same machinery becomes inconvenient or, in severe cases, dangerous. Biology often works through tools that are useful in one context and harmful in another. There are few simple heroes and villains inside the body. Context matters. Dose matters. Timing matters. Location matters. The immune system is not a collection of good cells defeating bad things. It is a network whose success depends on proportion. Perhaps that is why the immune system becomes more beautiful the longer we study it. At first, we imagine strength. Then we discover specificity, then memory, then communication, and eventually, we discover restraint. The mature immune system is powerful because it can act, but it is equally powerful because, most of the time, it chooses not to. Outside your window, perhaps the night has deepened. Maybe there is distant traffic. Maybe rain touches the roof. Maybe a fan turns slowly in the room. Or perhaps everything is completely still. Wherever you are, imagine the billions of human beings sleeping across Earth tonight. Some beneath city lights, some beside oceans, some in small villages surrounded by fields, some in apartments high above streets, some beneath mountains, some beside rivers. Each carries an immune system shaped by an ancient evolutionary story. And that story began long before humanity. Even simple multicellular organisms need ways to distinguish damaged tissue from healthy tissue and to respond to microbes. Innate immune mechanisms are ancient. Plants have sophisticated defenses too, though they work differently from the vertebrate immune system. In animals, immune strategies became increasingly varied over evolutionary time. The adaptive immune system found in jawed vertebrates introduced the extraordinary ability to generate highly diverse antigen receptors and form immunological memory. Long before mammals appeared, long before dinosaurs, long before forests looked anything like modern forests, the foundations of biological defense were already being written. Your immune system is therefore not a recent invention. It is an inheritance, a collection of solutions shaped and modified across immense stretches of time. Imagine Earth hundreds of millions of years ago. The continents are unfamiliar. The air feels different. Strange life moves through ancient seas. There are no human beings, no cities, no hospitals, no microscopes. Yet living organisms already face the same fundamental challenge: to remain themselves in a world filled with other life. Every organism is surrounded by chemistry, by competitors, by parasites, by microbes, by changing environments. Life needs boundaries, and life needs ways of repairing those boundaries when they fail. Evolution does not plan ahead. It works through variation and selection over generations. Useful biological traits tend to persist when they improve reproductive success. Over immense time, defense systems became layered. New mechanisms appeared upon older ones. Some were modified, some were lost, some became specialized, and eventually, after an almost unimaginable sequence of generations, there is you, lying quietly in the dark, carrying ancient cellular strategies within your blood. Your neutrophils do not know this history. Your macrophages have no awareness of evolution. A T cell does not understand why it has a receptor, yet each behaves according to molecular rules shaped by that past. That is one of the strange truths of biology. Wisdom can exist without thought. A spider does not study engineering before building a web. A seed does not understand spring before opening. Your immune system does not need to understand protection in order to protect you. It simply follows the deep logic written into living systems. And now we return to the present, to this breath. Air enters, air leaves. The heart continues its quiet rhythm. Blood passes through the lungs. Oxygen enters red cells. Immune cells drift beside them. Lymph moves slowly through vessels. Bone marrow continues making new cells. The spleen filters blood. The skin renews itself. The intestine maintains its crowded ecosystem. The thymus, whatever your age, carries the history of T cells educated there. And scattered through tissues are memory cells carrying traces of encounters from your past. Nothing needs your attention. Nothing needs your supervision. You can rest. Somewhere inside a lymph node, a lymphocyte may be moving slowly through a microscopic corridor. Somewhere in the skin, a macrophage may be clearing a dying cell. Somewhere in the lungs, cilia are sweeping mucus upward. Somewhere in the intestine, IgA is binding to a microbial surface. Somewhere in your bone marrow, a stem cell is dividing. Tiny events, ordinary events, events that have happened millions of times before. And together, they make up the quiet work of staying alive. So allow the story to become softer now. Imagine yourself floating once more through the bloodstream. There is no destination, only movement. Red blood cells drift around you like smooth crimson boats. The vessel curves gently through warm tissue. Far ahead, the pulse of the heart creates a slow, repeating rhythm. A pressure wave, then quiet. Another wave, then quiet. You pass a white blood cell moving along the vessel wall. It does not hurry. There is no emergency tonight. It simply travels, watching, waiting, ready if needed. The vessel becomes smaller. The current slows. Soon you reach a capillary. The walls are impossibly thin. Beyond them are ordinary cells going about their nighttime work. Some are using oxygen. Some are repairing proteins. Some are preparing to divide. Some are communicating with their neighbors. The immune system moves among them like a quiet presence, not separate from the body, part of the body, born from it, educated by it, regulated by it, and devoted, in its blind biological way, to preserving the conditions that allow the whole organism to continue. There is something deeply reassuring about that. Protection does not always announce itself. The strongest wall may be the one you never notice. The most successful immune response may be the infection that never develops far enough for you to feel sick. The most effective macrophage may complete its work without producing a sensation. The most useful antibody may bind its target in complete silence. Your body contains countless successes you will never know about. And perhaps it is enough simply to know that they happen. For now, drift deeper into rest. The microscopic landscape begins to fade. The blood river grows darker. The lymphatic streams become quiet silver lines. The great library of immune memory closes its doors for the night, though the librarians remain inside. The marrow continues its gentle work beneath the shelter of bone. The skin holds the boundary. The lungs breathe. The intestine rests in its warm darkness. And everywhere, tiny guardians continue their patient watch. You do not need to stay awake with them. They have been doing this for your entire life. Before you knew their names. Before you knew what a cell was. Before you could speak, before your earliest memory, they were already there, building, learning, remembering, repairing, protecting. And as sleep comes closer, imagine the entire body from a distance, not as organs and diagrams, not as labels in a textbook. See it instead as a landscape at night. The skin is the distant coastline. Blood vessels are rivers. Lymphatic vessels are quiet streams beneath moonlight. Lymph nodes are small lantern-lit stations along the road. The spleen is a harbor. The bone marrow is a warm workshop hidden beneath the hills. The thymus is an old schoolhouse where generations of young cells once learned the difference between vigilance and restraint. And throughout the countryside, countless cells move softly beneath the stars. Most of the land is peaceful. Most of the roads are empty. The gates remain open to ordinary life. A breeze crosses the fields. The river continues toward the sea. And somewhere far away, the first hint of morning is still many hours from arriving. There is time, plenty of time. Time for the body to rest. Time for cells to renew. Time for memories to settle. Time for the immune system to continue its ancient, invisible work. So you can let go of the day now. Let the details disappear. You do not need to remember neutrophils. You do not need to remember macrophages. You do not need to remember antibodies, complement proteins, lymph nodes, dendritic cells, B cells, or T cells. The body remembers enough. The body knows the routine, breathing in and slowly breathing out, while the quiet rivers continue flowing through the darkness. And somewhere within those rivers, a single white blood cell drifts onward, around one gentle bend, then another. Beneath the soft and endless night, the quiet river carries us onward. There is no hurry now. The bloodstream curves through the darkness of the body, branching into smaller vessels, then smaller ones still. Around us, red blood cells glide past in enormous numbers, carrying oxygen toward distant tissues. Between them, far less numerous but no less important, immune cells continue their patient journeys. Some are searching, some are returning home. Some have never encountered the particular thing they were created to recognize, and some carry memories from years ago. As we drift with them, another remarkable part of the immune story begins to appear. Because protection is not simply about recognizing an unfamiliar visitor. It is also about communication. Imagine that somewhere beneath the skin, a tiny group of cells has detected something unusual. Perhaps a microorganism has crossed a small break in the surface. The first cells to notice do not have voices. They cannot shout. They cannot ring bells. Instead, they speak chemistry. They release signaling molecules into the surrounding tissue. Among the most important families of these molecules are cytokines. Cytokines are small proteins that allow cells to influence one another. Some encourage inflammation. Some help immune cells grow or become activated. Some influence movement. Others help quiet a response once it is no longer needed. There are many different cytokines, and their effects depend on the cells involved and the surrounding conditions. So rather than imagining one alarm spreading everywhere, picture a collection of tiny messages moving through a neighborhood. A lantern appears in one window, then another. A messenger crosses a bridge. A gate opens. A patrol changes direction. Information moves outward from the place where something has changed. One group of signaling molecules is known as chemokines. Chemokines can help guide immune cells through tissues. Cells detect differences in chemical concentration and move toward stronger signals. Imagine walking through a dark forest while following the scent of wood smoke. At first it is faint. You continue forward. The scent becomes stronger, a little farther, stronger still. Eventually, you find the campfire. Immune cells can follow chemical gradients in a somewhat similar way. The process is called chemotaxis. It allows cells circulating through the blood to find particular locations where they are needed. And so, in a body containing trillions of cells, a microscopic disturbance in one tiny region can recruit help from elsewhere. No map is required. No conscious intelligence directs the journey. Only chemistry. We follow one neutrophil now. It is traveling through a small blood vessel. Nearby tissue has released signals associated with inflammation. The cells lining the vessel wall respond. Their surfaces change. The neutrophil begins interacting with them. At first, it slows. Then it rolls along the vessel wall. Molecular interactions become stronger. Finally, the neutrophil stops. Then something remarkable happens. It begins squeezing between the endothelial cells that form the vessel lining. The shape of the neutrophil changes. It presses through a space that appears almost impossibly narrow. And then, it is outside the bloodstream. Now it moves through tissue, following chemical signals. Closer, closer, until it reaches the region where help is needed. This process happens without conscious direction. Your mind may be occupied with a conversation, a meal, a song, or a dream, while a white blood cell quietly leaves a blood vessel somewhere inside you and finds its way toward a microscopic problem. Neutrophils are often among the earliest immune cells recruited to certain kinds of infection. They can engulf microbes. They can release antimicrobial substances. They can also form structures known as neutrophil extracellular traps. In this process, material containing DNA and antimicrobial proteins can be released into the surrounding space, where it may help trap microorganisms. It is an unusual strategy, a cell using part of its own internal material to create something like a microscopic net. Yet, as with many immune mechanisms, such responses must be controlled. A powerful defense can damage surrounding tissue if it becomes excessive. Again, balance. The same theme follows us everywhere. After some time, many neutrophils die. Their remains must be cleared. Macrophages arrive and engulf dying cells and debris. Signals begin to change. The inflammatory environment can gradually shift toward resolution. This stage is easy to overlook. We often think about how immune responses begin, but ending them is just as important. Once a threat has been controlled, the body needs to return toward ordinary life. Blood vessels settle. Chemical signals fade. Cells leave or die. Damaged structures are repaired. The neighborhood becomes quiet again. A successful immune response is not one that stays powerful forever. It is one that knows how to stop. Imagine a summer storm passing over a small town. For a while, rain fills the streets. Wind moves through the trees. People close their windows. Water rushes along gutters. Then the clouds pass. The wind softens. Drops fall more slowly from the leaves. Doors open again. The streets dry. Soon there is almost no sign that the storm was ever there. Inflammation often follows a similar arc. Beginning, escalation, control, resolution, repair, and then ordinary life. Now let us drift toward another organ. We pass through warm tissue and enter the liver. The liver is usually discussed as part of digestion and metabolism. And indeed, it performs an astonishing number of metabolic tasks. But it also contributes to immunity. Blood arriving from the digestive tract carries nutrients, microbial products, and countless molecules absorbed from the intestine. Much of this blood passes through the liver before returning to the general circulation. So the liver occupies a strategic position. It must examine material arriving from the gut while avoiding unnecessary reactions to the enormous number of harmless molecules associated with food and normal intestinal microbes. Within the liver live specialized macrophages known as Kupffer cells. They line spaces through which blood slowly flows. Imagine them resting beside a broad river. They watch what passes: old cellular material, fragments of microbes, particles carried from the intestine. Much is quietly removed. Much is tolerated. Once again, the immune system is making decisions without consciousness. Respond, ignore, clear, wait. The liver also produces many proteins that participate in immune responses. Some belong to the complement system. Others are known as acute phase proteins. During significant inflammation, the concentrations of certain proteins in the blood can change. One familiar example is C-reactive protein, often shortened to CRP. Doctors sometimes measure CRP because elevated levels can provide evidence that inflammation is occurring somewhere in the body. CRP does not usually tell us exactly what caused the inflammation. It is more like smoke visible above distant hills. It tells us that something may be happening. The details require further investigation. Inside the body, however, these proteins are not merely laboratory markers. They participate in the biological response itself. We leave the liver and continue through the circulation. Soon the current brings us toward the kidneys. The kidneys are not usually thought of as immune organs either. Their primary work involves filtering blood, regulating fluid and electrolytes, helping control blood pressure, maintaining acid-base balance, and performing several hormonal functions. But no organ exists outside the immune system's reach. Immune cells travel through kidney tissue. Resident cells can participate in local immune signaling, and the kidneys help maintain the internal environment in which every immune cell must function. This interconnectedness appears everywhere. The immune system depends upon circulation. Circulation depends upon the heart. Cells depend upon oxygen from the lungs. Protein production depends upon nutrients. Immune regulation is influenced by hormones. Hormones are influenced by sleep and circadian rhythms. The boundaries between systems are useful for textbooks. Inside you, the boundaries are far less obvious. Perhaps instead of thinking of your body as separate systems, we might imagine a forest. A forest has trees, soil, fungi, rain, insects, birds, rivers, microorganisms, sunlight. No single part is the forest. The forest exists through relationships. Your body is much the same. The immune system cannot be separated completely from everything else because immunity is woven through nearly every tissue. Even your nervous system communicates with immune cells. And this relationship becomes especially interesting when we consider fever. Suppose the immune system detects an infection. Certain immune signals can eventually influence the brain. In response, the hypothalamus can adjust the body's temperature set point. The result may be fever. A fever is not simply the body overheating accidentally. It is often a regulated physiological response. A moderate increase in body temperature can influence both microbial growth and immune activity. But fever also requires energy. It changes metabolism. It can make you feel tired. It encourages rest. Think back to the last time you had an ordinary infection. Perhaps you wanted nothing more than a bed, a blanket, a quiet room. Your muscles felt heavy. Your interest in the outside world faded. These sensations are sometimes called sickness behavior. They arise partly through communication between the immune system and the brain. The body is changing priorities. Activity becomes less important. Recovery becomes more important. Millions of years before humans invented hospitals, animals already responded to illness by becoming less active. Rest is ancient medicine, not a cure for every disease, not a substitute for medical care when it is needed, but an ancient part of the body's response to physiological stress. So when illness makes the body ask for stillness, there is biological history behind that request. Tonight, however, imagine the immune system in its ordinary state. No fever, no major disturbance, only routine maintenance. This may actually represent most of its life. The dramatic moments receive attention, but immune cells spend enormous amounts of time doing ordinary work, removing old cells, inspecting tissue, maintaining barriers, communicating with neighboring cells, watching microbial communities, clearing debris, waiting. It is less like an army constantly fighting, and more like a city maintenance department that also happens to operate an extraordinary emergency service. Most days are about keeping things orderly, and one of the largest maintenance tasks involves your own cells. Cells do not live forever. Some survive for days, others for months. Some can remain for many years. Every day, enormous numbers of cells reach the ends of their useful lives. Many undergo a controlled process known as apoptosis. Apoptosis is sometimes described as programmed cell death, but that phrase can sound more dramatic than the process usually is. Imagine a small shop in a quiet town reaching the end of its lease. The owner closes carefully. Shelves are emptied. Windows are covered. Everything is packed into neat containers. Then the building is dismantled without disturbing the neighborhood. During apoptosis, cells break themselves down in an organized way. Their contents are packaged. Signals appear on their surfaces. Phagocytic cells recognize those signals. Then the remains are engulfed and recycled. Usually there is little inflammation, no chaos, no spill, just removal. This happens inside you constantly. The body you have tonight is not exactly the body you had years ago. Cells have come and gone. Proteins have been replaced. Tissues have remodeled themselves. And throughout this turnover, immune cells help keep the landscape clean. Macrophages are especially important here. They do not merely respond to outsiders. They tend the body itself. A gardener does not spend every day chasing pests. Most days are spent pruning, clearing, turning soil, watching new growth. Now let us travel to the spleen again. This time we enter a region known as the red pulp. Here, old red blood cells are examined. A healthy red blood cell is flexible. It must squeeze through narrow spaces as it circulates. But as red cells age, their membranes become less flexible. Eventually, some can no longer pass easily through the spleen's filtering structures. Macrophages recognize and remove them. Their useful components are recycled. Iron from hemoglobin can be recovered and used again. The body wastes very little when it can avoid doing so. An atom of iron inside a red blood cell today may have existed in other cells before. Matter moves through you, reused, rearranged, returned. There is something soothing about that thought. Your body is not constantly building itself from completely new material. It is also recycling, repairing, reorganizing. The immune system participates in that quiet economy. Nothing dramatic, only careful housekeeping at microscopic scale. From the spleen, we drift toward the bloodstream once more. A B cell passes nearby. It has never encountered the antigen its receptor fits. Perhaps it never will. There are many such cells. The immune system maintains enormous diversity because the future cannot be predicted. No organism knows what microbial challenges tomorrow will bring. So evolution created a system based partly on preparedness through diversity. It is like keeping an immense collection of blank keys. Most will never be used. But if a strange lock appears, perhaps somewhere in the collection is a key that fits. This diversity arises through a fascinating rearrangement of DNA. During B cell development, sections of genes encoding antibody receptors are selected and joined in different combinations. Additional variation appears where these segments meet. The result is a tremendous range of possible receptor structures. T cells use a related strategy to generate their own receptor diversity. This process is unusual because most cells preserve their DNA sequence carefully. But developing lymphocytes deliberately rearrange particular regions of their genomes. A small controlled act of genetic creativity. One cell receives one arrangement. Another receives something different. Another, different again. Soon there is a population capable of recognizing an almost unimaginable range of molecular shapes. Of course, diversity creates danger too. Some randomly generated receptors could recognize the body's own molecules. So developing lymphocytes are subjected to processes that help remove or control strongly self-reactive cells. For B cells, much of this early screening occurs in the bone marrow. For T cells, as we discovered earlier, critical selection takes place in the thymus. Not every potentially self-reactive cell is eliminated. Additional regulatory mechanisms operate throughout the body. Among the most important are regulatory T cells. These cells help suppress inappropriate immune activation and maintain tolerance. Imagine them as quiet moderators in a crowded hall. Many voices are speaking. Some grow louder. The regulatory cells help prevent the conversation from becoming destructive. Again, strength through restraint. And now another immune cell approaches. An eosinophil. Eosinophils are less common in the blood than neutrophils. They participate in immune responses involving certain parasites and are also associated with allergic inflammation. Under a microscope, their granules take up particular laboratory dyes, which helped give them their name. Basophils are rarer still. They too contain granules and can participate in allergic and inflammatory responses. The immune system has many specialized characters. Some are common, some rare, some remain in tissues, some circulate, some live briefly, some persist. The diversity of cells mirrors the diversity of problems a living organism may encounter. Consider parasites. For much of human evolutionary history, parasitic worms and other organisms were common challenges. A large parasite presents a very different problem from a bacterium. A macrophage cannot simply swallow a worm that may be many times larger than itself. So the immune system developed other strategies. Antibodies such as IgA can participate. Eosinophils can release substances from their granules. Mast cells can alter local tissue environments. The response may affect mucus production and muscle contraction in ways that help remove certain parasites. Different problem, different solution. Biology is full of this kind of improvisation. Now imagine another visitor. A virus, much smaller, unable to reproduce independently. Its challenge is completely different. A virus hides inside cells, so antibodies can help block viral particles before they enter cells, while T cells and innate mechanisms become important once infection has begun inside tissues. Then imagine a bacterium living outside cells. Different again. Complement may help. Neutrophils may arrive. Antibodies may coat the bacterial surface. Phagocytes may engulf it. Then imagine a fungus. Again, the immune response changes. There is no single immune reaction. There are families of responses shaped to different biological situations. The immune system is less like one weapon and more like an entire language. Different sentences for different circumstances. And like language, immune responses depend upon context. A word spoken softly means something different from the same word shouted. A cytokine released in one tissue may have different consequences from that cytokine appearing somewhere else. A receptor signal combined with one set of additional signals may activate a cell. The same receptor signal without proper support may lead to silence or tolerance. Context. Always context. We continue drifting. The body is warm around us. The outside world is now very far away. Perhaps you can hear only the quietest sounds in your room. And somewhere behind those sounds is your breathing. In and out. In and out. Every breath brings oxygen. Every breath also exposes your respiratory surfaces to the environment. Yet most breaths pass without incident. You have taken hundreds of millions of breaths over a lifetime. Almost all of them were completely ordinary. That may be one of the immune system's greatest achievements. Ordinary life remains ordinary. Now let us imagine one breath entering through the nose. The air is cooler than the tissues inside. It moves past tiny hairs near the entrance. Farther in, it flows across moist surfaces. The nasal passage is warm and humidified. Particles collide with mucus. Immune molecules wait within secretions. Some microbes become trapped. Then cilia gradually move the mucus toward the throat. Nothing dramatic occurs. No symptoms appear. You never know that anything happened. Protection has succeeded invisibly. This invisible protection extends to the eyes. Every time you blink, your eyelids spread tears across the surface. Tears keep the eyes moist. They also contain protective substances, including lysozyme, an enzyme capable of damaging certain bacterial cell walls. Blinking helps wash particles away. The tear drainage system carries fluid toward the nasal cavity. Even something as gentle as a tear participates in defense. Think about that for a moment. The human body protects itself not only with immune cells and antibodies, but with flowing water, tears, saliva, mucus, urine. Each can help move unwanted material away from vulnerable surfaces. Sometimes Sometimes the simplest defense is movement. Do not let something remain long enough to settle. The urinary tract uses this principle too. The flow of urine helps reduce the ability of microbes to remain within parts of the urinary system. Chemical conditions provide additional protection. Again, there is no single barrier. Several layers work together. Flow, chemistry, epithelial cells, innate immune signals, adaptive immunity, layer upon layer. We travel back toward the skin. A bead of sweat rests on the surface. Sweat helps regulate body temperature, but the skin's secretions also contribute to its chemical environment. Sebaceous glands produce oily substances known collectively as sebum. Skin microbes interact with these secretions. The result is an environment that favors some organisms more than others. Your skin is not a blank wall. It is a habitat. Different regions host different communities. The moist environment of an armpit differs from the dry surface of a forearm. The oily skin of the face differs again. Each microscopic neighborhood has its own conditions: temperature, moisture, oil, salt, acidity, and each supports a somewhat different microbial community. Imagine walking across these regions at microscopic scale. The landscape changes gradually. A dry plain becomes a humid valley. A hair follicle descends like a deep cave. Sebum coats its walls. Microbes live nearby. Immune cells remain beneath the epithelial surface. There is activity everywhere. And yet, from your ordinary perspective, it is simply skin, warm beneath a blanket, quiet in the night. Now we approach a hair follicle. The follicle extends down into the skin. Nearby are glands and tiny blood vessels. Immune cells can gather around follicles when needed. The skin contains specialized immune populations, including dendritic cells and tissue-resident T cells. Some immune cells remain in tissues for long periods rather than constantly circulating through blood. These resident cells can provide rapid local surveillance. They know this neighborhood in a biological sense. They have been here before. Tissue-resident memory T cells are particularly fascinating. After certain infections, some memory T cells remain within tissues such as skin or mucosal surfaces. If the same pathogen appears again in that region, these resident cells may respond rapidly. Imagine a village that once experienced a flood. Years later, some people who remember the old water line still live there. They know which roads disappeared first. They know where the river crossed the bank. They do not need to wait for instructions from a distant capital. They remember locally. Your tissues can carry local immune memories too. Memory changes everything. The first encounter with a pathogen can require time. Rare lymphocytes with appropriate receptors must be found. They must become activated. They must multiply. Specialized cells must develop. Antibodies may need to improve, but during later encounters, memory cells can respond much more quickly. This is why immune memory can make a second exposure very different from the first. The body has learned, and B cells can refine their antibodies during an immune response. Within specialized structures called germinal centers, often found in lymph nodes and the spleen, activated B cells can undergo processes that change their antibody genes. Cells producing antibodies that bind antigen more effectively can be preferentially selected. Over time, the average affinity of antibodies can increase. This is called affinity maturation. It is almost as if the immune system begins with a rough sketch, then slowly sharpens the lines. The first key fits. Later keys may fit even better. B cells can also change the class of antibody they produce through a process called class switching. The antigen specificity can remain similar while the antibody's functional properties change. A B cell lineage might begin by producing IgM and later produce IgG, IgA, or another class depending on the signals it receives. The recognizing end remains directed toward the same target, but the rest of the molecule changes what can happen after binding. Imagine keeping the same carefully shaped hand, while changing the tool attached behind it. Recognition remains. Function adapts. These processes take place in microscopic regions you will never see. A lymph node may be smaller than a bean, yet inside it, cells are migrating, dividing, competing for signals, exchanging molecular information, and refining antibodies. The complexity rivals a city, perhaps exceeds one, and there may be hundreds of lymph nodes distributed throughout the body. Tiny centers of biological conversation. Let us enter one again. This time, instead of focusing on individual cells, imagine the architecture. There are regions rich in B cells. Other areas contain many T cells. Lymph flows through channels. Dendritic cells display antigens. Macrophages capture material. Blood vessels allow lymphocytes to enter from the circulation. Cells constantly arrive and depart. A lymphocyte may enter one node, search for its antigen, find nothing, leave through lymphatic vessels, return eventually to the bloodstream, and enter another node somewhere else. Searching, always searching. Most searches end with nothing, and that is perfectly fine. Think of millions of librarians walking through millions of shelves. Each carries a single question. Have you seen this shape? No. They continue. Another shelf, another room, another node. Then, perhaps once in a great while, yes, the match appears. And suddenly the quiet cell changes. It stops wandering. Signals arrive. Genes switch on. The cell grows. It divides. Its descendants take on new roles. A rare recognition becomes a population. The immune system has found what it was looking for. But even then, the response is not permanent. After the problem is resolved, most activated cells disappear. Maintaining enormous armies forever would consume too much energy and increase the risk of unnecessary damage. So the population contracts. Only memory remains. A smaller group, enough to remember, not enough to burden the body. Economy again. Biology spends energy carefully. Energy matters enormously to immunity. Producing millions of immune cells requires nutrients. Making antibodies requires amino acids. Cell division requires energy. Fever increases metabolic demand. Tissue repair requires raw materials. This is one reason the body coordinates immunity with metabolism. During illness, appetite may change. Energy use shifts. Hormones influence immune cells. Immune signals influence metabolism. Nothing occurs independently. And sleep enters the story once more. As evening arrives, your internal clock changes the hormonal landscape. Melatonin rises in response to darkness under normal circadian conditions. Cortisol follows its own daily rhythm. Body temperature changes. Immune cell trafficking changes. Certain aspects of immune signaling follow daily patterns. Your body has temporal architecture. There is a time for wakefulness, a time for feeding, a time for activity, and a time for sleep. Tonight belongs to that last rhythm. Imagine your body as a city seen from high above after midnight. Most office lights are off. Roads are quieter. Factories have changed shifts. Maintenance crews move through empty streets. Waste is collected. Repairs happen while traffic is low. Hospitals remain open. Fire stations remain ready. The city is resting, but not inactive. Sleep is something like that. Conscious activity fades, yet physiology continues. The immune system does not sleep in the way your mind does. It changes rhythm. It keeps watch. We float now toward the brain. For many years, the brain was sometimes described as an immune-privileged organ, separated from ordinary immune activity by the blood-brain barrier. There is truth in the idea that the brain's immune environment is highly specialized. But the older picture of complete isolation was too simple. The brain has its own resident immune cells, microglia. These small cells extend branching processes into the surrounding neural tissue. They constantly survey their environment. They remove debris. They respond to injury and infection. They participate in the shaping and maintenance of neural connections. Imagine them as gardeners in an immense forest of neurons. They move quietly between branches, inspecting, pruning, clearing, watching. The blood-brain barrier still plays a crucial protective role. Specialized cells and tight junctions regulate what can move from the bloodstream into brain tissue. This protects the delicate chemical environment required for neurons to function. But communication still occurs between the nervous and immune systems. Signals pass in both directions. Immune activity can affect mood, fatigue, appetite, and sleep. Neural and hormonal signals can influence immunity. The old boundary between mind and body becomes difficult to maintain when we look closely enough. You are one organism. During infection, perhaps you have noticed how thinking changes. Concentration becomes difficult. Motivation fades. You want to lie still. The outside world seems less important. Part of this experience arises because immune signals influence the brain. The sensation of being sick is not simply the direct effect of microbes. It is also the body coordinating behavior with defense. Rest. Conserve energy. Avoid unnecessary activity. Give physiology time. Tonight, though, there is no need to imagine sickness. Instead, picture microglia moving quietly through healthy neural tissue. The neurons around them communicate through electrical and chemical signals. Memories are stored across networks. Dreams may soon begin. And through this astonishing landscape, immune-related cells continue their maintenance. Even the place where your dreams will unfold has guardians. Now we drift downward again, past the throat, past the heart, toward the intestine. The microbial city is still there. Billions upon billions of organisms living within the digestive tract. Some scientists have compared the microbiota to another organ because of its broad influence on metabolism and physiology. The comparison is imperfect, but it captures something important. These microbes are not merely passengers. They participate in the ecosystem of the body. They break down compounds human enzymes cannot completely digest. They produce metabolites that can influence intestinal cells. They compete with invading organisms, and they interact continuously with immunity. One group of compounds produced when certain gut bacteria ferment dietary fiber are short-chain fatty acids. These molecules can be used by intestinal cells and can influence immune and metabolic processes. Diet, microbes, intestinal barriers, and immunity are therefore connected through chemistry. A piece of plant material eaten hours ago, broken down by microbes, can become molecules that interact with your own cells. The boundary between environment and body is wonderfully porous. Food becomes chemistry. Chemistry becomes tissue. Tissue becomes you. And yet the immune system must maintain boundaries within this intimacy. The gut contains enormous numbers of bacteria. If those organisms routinely entered the bloodstream, serious problems could occur. So the intestinal barrier remains essential. Epithelial cells are joined closely. Mucus separates many microbes from the cell surface. Antimicrobial molecules are released. IgA binds microbes and antigens. Immune cells monitor what crosses. Several layers. Always several layers. Perhaps this is the central theme of immunity. There is rarely one guardian. Instead, there are communities. Skin cells work with microbes. Mucus works with cilia. Antibodies work with phagocytes. Dendritic cells work with T cells. T cells work with B cells. Bone marrow supplies the population. Blood carries it. Lymph connects tissues. The spleen filters. The liver participates. The brain communicates. Sleep regulates. No cell protects you alone. Protection is cooperation. And now, as the night becomes quieter still, let us imagine that cooperation from far away. Your body is no longer a collection of organs. It is a softly glowing landscape. Blood vessels shine like rivers beneath moonlight. Lymphatic vessels appear as smaller silver streams. Lymph nodes rest along them like warm houses in distant valleys. The bone marrow glows beneath mountains of bone. The lungs open and close like two quiet forests moving with the wind. The intestine winds through the landscape like a long, fertile valley filled with microscopic life. And around everything, the skin forms a gentle horizon. Beyond it is the world. Within it is you. A particle lands upon the skin. Nothing happens. A harmless bacterium enters the mouth. Nothing noticeable happens. Dust reaches the nose. Mucus catches it. A damaged cell signals for cleanup. A macrophage arrives. An old red blood cell reaches the spleen. It is recycled. A B cell enters a lymph node. Its antigen is not there. It leaves. A T cell passes through tissue. Everything appears normal. It continues. Thousands, millions, billions of quiet decisions, most ending with the same conclusion. All is well. Move on. Perhaps this is what makes the immune system so comforting to explore at bedtime. Its greatest talent is not fighting. Its greatest talent is maintaining peace. It watches so that you do not have to. It remembers so that your conscious mind does not need to. It repairs without being asked. It replaces what has been lost. And when a response is finished, it tries to become quiet again. You can become quiet too. There is nothing more you need to understand tonight. Let the names fade. Cytokines, chemokines, neutrophils, macrophages, lymphocytes, antibodies, memory cells. All of them can drift away like words written on water. The science remains true even when you stop thinking about it. The cells continue their work whether you remember their names or not. Your breathing slows. The chest rises gently, then falls. Air moves through the quiet passages of the lungs. Oxygen crosses into blood. The heart sends it onward. Somewhere beneath the ribs, the spleen waits beside the river. Deep inside the bones, marrow continues producing new cells. Along the intestine, microbes live beneath a protective layer of mucus. Across the skin, microscopic communities rest beneath the darkness of the room. And through every region, the immune system remains present. Not tense, not frightened, simply ready. Imagine now that you are standing on a hill overlooking that inner landscape. Night covers everything. Far below, the blood rivers move slowly through the valleys. Tiny lights mark lymph nodes in the distance. A warm glow comes from the marrow beneath the hills. There is no alarm, no urgency. Only the steady rhythm of a living world taking care of itself. A breeze moves through the imaginary trees. Far away, water passes over stones. The stars above are quiet, and somewhere along a narrow road, a single immune cell continues its patrol. It has no idea that you exist as a conscious person. It does not know your name. It does not know your memories. It does not know the sound of your voice. And yet, its entire existence is part of keeping you here. So let it continue. Let the rivers flow. Let the marrow work. Let the lungs breathe. Let the skin hold the boundary. Let the immune system keep its gentle watch. And you can simply sleep. And still, the story continues. Because the immune system is never truly finished. There is no final patrol, no last inspection, no moment when the body closes its gates and says that its work is complete. Life is movement. Cells age. New cells appear. Food enters. Air arrives. Microbes come and go. Tiny injuries heal. Old proteins are dismantled. New ones take their place. And through all of this change, the immune system continues adapting to the world around you. Imagine that the night has grown deeper. Outside, streets have become quieter. Windows are dark. Trees stand almost motionless beneath the sky. Perhaps a distant car passes now and then. Perhaps there is only silence. Inside your body, however, another journey is beginning. This time we will follow a single young immune cell. It begins far from the outside world, inside the protected darkness of your bone marrow. The marrow is warm, soft, rich with blood vessels. Around us, cells exist at many stages of development. Some will become red blood cells. Others will become platelets. Others are beginning the long process of becoming immune cells. At the center of this remarkable family tree are hematopoietic stem cells. A stem cell divides. One descendant maintains the stem cell population. Another may begin moving toward a more specialized future. With each developmental step, possibilities narrow. A cell that once had many potential identities gradually commits to one. Perhaps it will become a neutrophil, perhaps a monocyte, perhaps a lymphocyte. Tonight, we follow a cell destined for the adaptive immune system. A young lymphocyte. It has no memories yet, no experience. It has never encountered a virus, never recognized a bacterial protein, never traveled through a lymph node. Its world is only marrow. Inside this developing cell, DNA is being rearranged. Specific gene segments are selected. Pieces are joined. A unique receptor begins to emerge. The process contains an element of randomness, and that randomness is valuable. The body cannot predict every molecular shape that future pathogens might carry. So instead of trying to predict the future, it creates diversity. Millions of cells, millions of variations, an enormous collection of possibilities. Our young cell receives one receptor, only one among an astonishing range. Perhaps that receptor will someday recognize something important. Perhaps it never will. There is no way to know. And yet the body makes the cell anyway. Preparedness requires possibilities that may never be used. If our cell is destined to become a B cell, much of its early maturation occurs here in the bone marrow. If it belongs to the T cell lineage, its journey takes another path. Let us imagine this as a young T cell precursor. Soon it leaves the marrow. It enters the blood. For the first time, the current carries it beyond the place where it was born. Red blood cells surround it. The heart draws closer. The current accelerates. Then the cell is carried toward the chest, toward the thymus. The thymus is different from the marrow. Its architecture is carefully organized. Developing T cells enter and begin an extraordinary series of tests. The first challenge is simple in principle. Can the receptor function at all? T cells need to recognize antigens only when those antigens are displayed by particular molecules on the body's cells. A receptor that cannot interact appropriately with these display molecules will be of little use. Many developing T cells fail this test. They quietly disappear. There is no tragedy in this. Cell death is part of development. The body produces more possibilities than it ultimately needs. The cells that continue face another challenge. Do they react too strongly to the body's own molecules? Cells within the thymus expose developing T cells to a remarkable variety of self-antigens. Some of these antigens represent proteins normally produced in distant organs. Through specialized genetic regulation, thymic cells can display samples of proteins associated with tissues elsewhere in the body. It is as though the thymus contains a miniature catalog of self. A young T cell walks through this molecular gallery. Heart, skin, pancreas, liver, other tissues. Not literally, of course. Only molecular samples, but enough to test an important question. Will this T cell react dangerously against the body it is supposed to help protect? If the answer is yes, the cell is usually removed or diverted into specialized regulatory pathways. If the answer is no, it may continue. Imagine the thymus as a quiet school at night. Long corridors, soft light, rooms filled with molecular lessons. The young cells move through them. Most do not graduate. Those that do leave with something extraordinarily important. Not knowledge in the conscious sense, but biological restraint. They have been shaped to function within the body without strongly attacking ordinary self-tissues. Our T cell eventually reaches the end of this process. It is released. The bloodstream receives it. And now, its real journey begins. The cell travels through blood, then through lymphoid tissues. It enters a lymph node. It searches. Nothing. It leaves. Days pass. Another lymph node. Nothing. Then another. Still nothing. Weeks may pass. Months. The cell continues circulating. Perhaps years. Its receptor remains unchanged. It waits for the molecular shape it was created to recognize. Most of its existence is patience. Meanwhile, life continues outside. You wake in the morning. You eat breakfast. You walk beneath sunlight. You speak with people. You touch doors. You breathe the air of rooms and streets. You laugh. You worry. You work. You sleep. And the T cell knows none of this. It simply circulates, searching. Then one ordinary day, somewhere in the body, a dendritic cell encounters material associated with a microorganism. The dendritic cell processes proteins from what it has encountered. Small fragments are placed on display molecules at its surface. The dendritic cell travels toward a nearby lymph node. There it settles among thousands of passing T cells. One approaches. No match. Another. No match. Another. Nothing. Then our cell arrives. Its receptor touches the molecular display. And after perhaps years of waiting, the shapes fit. The contact is microscopic. No sound. No flash of light. Yet for this cell, everything changes. Additional signals confirm that a response is appropriate. Internal pathways activate. Genes that had remained quiet become active. The cell begins producing new proteins. Its metabolism changes. It grows. Then it divides. 1 becomes 2. 2 become 4. 4 become 8. The population expands. The rare cell is rare no longer. A receptor that existed in only a tiny number of cells is now carried by many descendants. The body has transformed recognition into numbers. Some descendants become active effector cells. Others will eventually become memory cells. Depending on the type of T cell and the signals present, their roles differ. Some help B cells. Some activate or coordinate other immune cells. Some can recognize infected cells. Each performs a particular part of the larger response. But notice what has happened. The immune system did not know in advance that this particular T cell would be useful. It simply maintained diversity, waited, and when the right situation appeared, it expanded what worked. This principle is called clonal selection. It is one of the foundations of adaptive immunity. Now imagine a B cell elsewhere in the lymph node. Its surface antibody recognizes a structure from the same microorganism. The B cell binds the antigen. It takes some of that material inside, processes it, then displays fragments on its own surface. A helper T cell recognizes the displayed fragment. The two cells communicate. Signals pass between them. The B cell receives permission, in a sense, to proceed. It divides. Its descendants begin changing. Some become plasma cells, and suddenly antibodies begin pouring into the circulation. A plasma cell looks different from the resting B cell it once was. Its interior becomes packed with machinery for producing proteins. The endoplasmic reticulum expands. The cell becomes a biological factory. Antibody molecules are assembled, folded, processed, released, again and again and again. A single plasma cell can produce large quantities of antibody. Now multiply that by thousands of plasma cells, then millions. Soon the blood and tissue fluids contain enormous numbers of molecules shaped to recognize the same target. An antibody drifts through the bloodstream. It has the familiar Y-like structure often shown in diagrams. At the tips of the Y are regions capable of binding antigen. These regions contain the extraordinary molecular variation generated during B cell development. The lower portion of the antibody interacts with other parts of the immune system, so an antibody performs two broad tasks: recognition at one end, communication and biological action at the other. It is a bridge. One antibody encounters its target. It binds. Another binds nearby. Soon the surface is coated. This coating may make the target easier for phagocytes to recognize. Macrophages and neutrophils carry receptors capable of interacting with the constant regions of certain antibodies. The phagocyte does not need to independently solve the entire recognition problem. The antibody has already marked the target. Imagine walking through darkness and seeing small reflective markers placed along a path. The road becomes obvious. Antibodies can provide something similar for immune cells. They turn recognition into visibility. Other antibodies can neutralize. Suppose a virus needs to attach to a particular receptor on a human cell before it can enter. An antibody binds to the part of the virus needed for that attachment. The virus approaches the cell, but its molecular key is covered. It cannot easily use the receptor. The infection chain may be interrupted before it begins. No dramatic destruction is necessary. The door simply remains closed. This is one of the gentlest immune strategies. Do not destroy. Do not attack. Simply block. Sometimes protection is nothing more than preventing two molecules from touching. Eventually, the immune response reaches its peak. Then the microorganism declines. Antigen becomes less abundant. Inflammatory signals weaken. The body no longer needs such a large population of activated cells. Most effector cells die. Antibody levels may gradually decrease. The lymph node becomes quieter, but a few descendants remain. Memory cells. Our original T cell may now exist through a population of descendants scattered through the body. Some circulate. Some remain in lymphoid organs. Some may settle in tissues. The original encounter has changed the future. Years may pass. The person grows older. Perhaps moves to another city. Perhaps changes careers. Perhaps has children. Perhaps forgets the illness entirely. But somewhere inside, memory cells remain. Then the same pathogen appears again. This time the search does not begin from the beginning. Memory cells are more numerous than the original rare cells were. They can respond quickly. B-cell memory can rapidly generate new antibody-producing cells. Existing antibodies may already be present. The second response can therefore be faster and stronger, sometimes so effective that the person barely notices the encounter. The immune system remembers something the conscious mind has completely forgotten. Your body is filled with these invisible histories. Every scar has a story. Every memory cell has a story. Every antibody lineage was shaped by some encounter or immunization. Some stories began in childhood. Some began recently. Some may have started so early that you have no conscious memory of the time. Biology remembers in ways that the mind cannot. Now let us travel backward through human history. Long before anyone knew that memory cells existed, people noticed the effect. Ancient observers recognized that survivors of some diseases were less likely to suffer the same illness again. They did not know why. There were no microscopes capable of showing lymphocytes, no knowledge of antibodies, no concept of viruses. Yet the pattern was there. Nature had revealed the answer before science understood the mechanism. Centuries passed. Microscopes improved. Scientists discovered cells, then microorganisms. Medicine slowly began connecting particular microbes with particular diseases. By the late 19th and early 20th centuries, immunology began emerging as a recognizable scientific field. Researchers studied antitoxins, antibodies, complement, phagocytosis, cellular immunity. Each discovery revealed another part of a system far more complex than anyone had imagined. One of the great early debates involved different ways of thinking about immunity. Some scientists emphasized soluble factors in the blood. Others focused on cells that engulfed microbes. Over time, it became clear that both perspectives contained important truths. Antibodies matter. Phagocytes matter. Complement matters. T cells matter. The immune system is not one mechanism. It is collaboration. Again and again, biology refuses simple answers. Imagine an old laboratory late at night. Glass bottles stand on wooden shelves. A microscope rests beneath lamplight. Outside, horses move quietly in a stable. Researchers write observations by hand. They do not yet know about DNA. They have never seen the molecular structure of an antibody. The word cytokine is far in the future, yet they are beginning to uncover the hidden rules of protection, one observation at a time. Decades later, scientists would discover that antibodies are proteins. Then their structures became clearer. Researchers learned that different lymphocytes carry different receptors. The idea of clonal selection developed. T cells and B cells were distinguished. The major histocompatibility complex was explored. Monoclonal antibody technology arrived. Genes involved in immune receptor diversity were uncovered. Each discovery opened another door. And behind every door, more complexity. Today, researchers can watch immune cells moving through living tissues using sophisticated microscopy. They can sequence the receptors of individual lymphocytes. They can measure thousands of genes becoming active during immune responses. They can trace the ancestry of antibody-producing cells. They can study how immune populations differ between tissues. Yet even now, the immune system contains mysteries. Why do some people develop particular autoimmune diseases while others do not? Why do immune responses vary so much between individuals? How does the microbiome influence immunity across a lifetime? Why does immune memory last decades in some situations and fade more quickly in others? How can immunity be guided to recognize cancer more effectively? These questions continue. Science has learned an enormous amount, and there is still more to discover. Cancer brings us to another remarkable role of immunity. Your cells divide throughout life. Whenever DNA is copied, errors can occur. Cells also accumulate genetic changes from environmental exposures and ordinary biological processes. Most mutations do not produce cancer. Many are harmless. Some damaged cells die. Others are controlled by internal mechanisms that prevent inappropriate growth. But occasionally, a cell acquires changes that allow it to grow abnormally. The immune system can sometimes recognize these altered cells. This process is part of what researchers call immune surveillance. A cancer cell is difficult for the immune system because it began as self. It is not an invading bacterium. It is one of the body's own cells that has changed. Some tumors display abnormal proteins or altered patterns that immune cells can recognize. Cytotoxic T cells may attack such cells. Natural killer cells can respond to certain abnormalities. But cancers can also evolve ways of avoiding immune responses. Some reduce the signals that make them visible. Some create environments that suppress immune cells. Some exploit natural regulatory pathways that normally prevent excessive immune activity. It becomes a complex biological conversation. Modern cancer immunotherapy grew from understanding these conversations. Some treatments help remove molecular breaks that tumors exploit to suppress T cells. Others use engineered antibodies. Some approaches collect a patient's immune cells, modify or expand them, and return them to the body. The details can become very technical. Tonight, we need only appreciate the larger idea. Medicine is learning how to cooperate with the immune system, not simply replacing it, not commanding it, but understanding its language well enough to guide it. The same is true in transplantation. The same is true in vaccines. The same is true in treatments for autoimmune disease. Much of modern immunology is the science of adjustment. Increase a response here, reduce one there, create memory, restore tolerance, block a signal, strengthen recognition. The immune system is powerful precisely because it is regulated. Medicine often works by changing that regulation carefully. Now drift away from the laboratory, away from hospitals, back into the quiet body. Because tonight we are not here to think about treatment. We are here to appreciate ordinary protection, the kind happening right now. A macrophage rests within your liver. A dendritic cell waits beneath a barrier surface. A memory T cell sits quietly in tissue. A B cell travels through a lymph node. A neutrophil circulates through blood. A natural killer cell inspects neighboring cells. Bone marrow produces replacements. Old immune cells disappear. New ones enter. The population changes every moment. And still, the system remains recognizably you. Age changes immunity. A newborn's immune system is not identical to that of an adult. Before birth, antibodies from the mother can cross the placenta and provide temporary protection. After birth, antibodies in breast milk, particularly secretory IgA, can contribute protection at mucosal surfaces when breastfeeding occurs. Meanwhile, the infant's own immune system begins encountering the microbial world. The microbiota develops. Vaccinations can introduce controlled immune lessons. Lymphocyte populations change. The immune system matures through experience. Childhood is therefore a period of extraordinary immune education. A child touches the floor, touches toys, explores the world, breathes air filled with particles, eats new foods, encounters microbes. The immune system receives an endless stream of information. Most of it becomes ordinary. Some creates memory. Some helps shape tolerance. Gradually, the immune landscape becomes richer. Then adulthood arrives. The thymus becomes less active than it was during childhood, though T cell populations continue to function. Memory accumulates. Years of encounters leave their traces. Later in life, the immune system changes again. This broad process is often called immunosenescence. Some immune responses become less efficient. The composition of immune cell populations shifts. Chronic low-level inflammation can become more common with aging. Yet older immune systems also contain decades of accumulated memory, a lifetime written into cells. Imagine an elderly person sleeping peacefully, perhaps 80 years old. Inside that body are immune memories shaped across most of a century. Childhood infections, vaccinations, seasonal viruses, environmental exposures, countless encounters that were never consciously noticed. The cells carry a biological autobiography. No pages, no photographs, only molecular memory. Your own immune autobiography is being written tonight, not necessarily through some dramatic event, perhaps only through ordinary maintenance. A memory cell survives another day. A new lymphocyte leaves the marrow. A macrophage clears debris. A barrier remains intact. Small things. But a life is made from small things repeated for years. And perhaps this is a good place to become even quieter. The science can begin fading into scenery. Imagine the bone marrow again. A warm cavern beneath the hard shelter of bone. Tiny new cells appear like lights in the distance. Some remain, some depart. The blood carries them away. Follow one vessel outward. It joins another. The current grows. Soon we reach the heart. The chambers open and close. Valves move. Blood flows forward. The rhythm is steady, not perfectly identical from beat to beat. Living rhythms rarely are, but steady enough, reliable enough, a pulse you have carried since before birth. The blood enters the lungs. Oxygen arrives, then back to the heart, then outward again, past the liver, past the kidneys, through the brain, into muscles, into skin, through every region of the body. And among the red cells travel the guardians we have met. Neutrophils, monocytes, lymphocytes, eosinophils, basophils, different travelers sharing the same river. Then some leave the river. They enter tissue. Others enter lymph nodes. Others settle permanently in particular organs. Every cell has its route. Every route is guided by molecular signals. No road signs, no maps, no central traffic controller. Yet somehow the system works. Imagine standing beside one of those microscopic roads at midnight. A lymphocyte passes, then another. A macrophage remains beside the tissue. Farther away, an endothelial cell lines a blood vessel. The surrounding cells exchange quiet chemical messages. Nothing urgent, only conversation. The biological equivalent of lights glowing behind windows in a sleeping town. The town does not need you tonight. The roads know where to go. The gates know when to open. The river knows how to flow. The marrow knows how to make new cells. The lymph nodes know how to organize their populations. The lungs know how to breathe. And your immune system knows how to continue the work it has been doing since long before you learned its name. So imagine yourself leaving the microscopic world, slowly rising. The cells become smaller beneath you. Blood vessels become thin red lines. Organs return to their familiar shapes. The body becomes whole again. Then even that image fades. There is only the room, the pillow, the blanket, the soft weight of your body resting against the bed. Your skin is warm. Your breathing is easy. The jaw can loosen. The forehead can become smooth. Your hands do not need to hold anything. Your feet do not need to carry you anywhere. There is nothing to solve. Nothing to remember, nothing to prepare. The immune system will keep watch. The heart will keep beating. The lungs will keep breathing. The marrow will keep building. the lymph will keep moving, and you can move in the opposite direction, away from wakefulness, away from thought, towards sleep. Imagine one final lymph node in a quiet valley. Moonlight lies across the hills. A narrow stream moves beside it. Inside, the last few travelers of the evening pass through. One cell enters, another leaves. No alarms, no urgency, only watchfulness. Far beyond the valley, the lights of the immune system stretch into the distance. One by one, they seem to grow softer, not because they have stopped working, but because you are drifting farther away. The science becomes a dream. The bloodstream becomes a river. The lymph nodes become small houses beside the water. The marrow becomes a warm light beneath the earth. The lungs become forests moving slowly in the wind. And the skin becomes the distant shoreline of a peaceful country. Everything within that country continues its quiet work. Everything is moving gently. Everything has time. And somewhere, almost too small to imagine, a memory cell waits. It may wait for years. It asks for nothing. It requires no conscious thought. It simply carries a tiny piece of biological history into tomorrow. And while it waits, the river flows past, slowly, quietly, endlessly, through the warm darkness of the sleeping body. The river carries us farther into the night. And now, perhaps, we can leave individual cells behind for a while. Instead, imagine the immune system as a whole, not as a collection of separate organs, not as a diagram, but as a living network spread through nearly every corner of you. A network that changes with the hour, with your age, with the places you have lived, with the microbes you have encountered, with the food you have eaten, and even with the rhythm of waking and sleeping. Your immune system is not exactly the same today as it was 10 years ago. It has a history. And tonight, we will drift slowly through that history. Long before you were born, your immune story had already begun. Before your first breath, before your first glimpse of daylight, you developed within a protected environment inside your mother. Your own immune cells began forming before birth. Your blood developed. Your thymus developed. Lymphoid tissues began taking shape. But you were not completely alone in your protection. During pregnancy, maternal IgG antibodies can cross the placenta. These antibodies provide a newborn with a temporary inheritance of immune protection, not genetic inheritance, not permanent memory, something more fleeting. Antibodies borrowed for the beginning of life. Imagine a quiet bridge between two generations. Across that bridge move molecules created by one immune system, entering another. A mother may have acquired those antibodies through past infections or vaccination. Now, for a limited time, some of that protection is shared. After birth, the transferred antibodies gradually decline. The infant's own immune system increasingly takes responsibility. But for those first vulnerable months, the borrowed protection can matter. It is one of the gentlest beginnings in biology. Before the newborn immune system has learned much about the world, it receives a small collection of lessons from someone who arrived earlier. Then comes birth. For the first time, the infant enters an environment filled with microorganisms. Air touches the lungs. Skin meets clothing. Hands touch faces. Milk enters the digestive system. Microbes begin colonizing the skin and intestine. The immune system starts meeting the wider world, not as an enemy, as a new environment. The early microbiome develops, through a complicated combination of factors. Birth circumstances can influence it. Feeding influences it. Environment matters. Antibiotic exposure can matter. Family members contribute microbes. Pets may contribute. Food later changes the ecosystem again. There is no single ideal microbial community shared identically by every healthy person. Human microbiomes vary. They are personal ecosystems, and like forests, they change over time. Imagine the infant intestine during those early days. At first, the landscape is relatively new. Then microorganisms arrive. Some find suitable conditions. They multiply. Others disappear. Different species interact. The chemistry of the intestine changes. The immune system observes all of it. This is not simply infection prevention. It is education. Immune cells learn to tolerate ordinary food molecules. They encounter harmless microbes. They develop regulatory patterns. The barrier matures. A relationship begins between human tissue and microbial life that may continue for decades. If the infant receives breast milk, another layer of biology enters the story. Human milk contains nutrients, of course, but it also contains antibodies, particularly secretory IgA, along with many other biologically active components. Secretory IgA can help protect mucosal surfaces without producing the kind of strong inflammatory response that would be undesirable in a developing intestine. Again, protection without unnecessary disturbance. The same theme. As infancy becomes childhood, the immune system's library expands. A child crawls across floors, touches toys, puts fingers into a mouth, runs outdoors, breathes pollen, plays in soil, meets other children. Every day brings molecular information. Most encounters are harmless. Some cause infection. Some become immune memories. Vaccinations add carefully designed lessons. And gradually, the immune system becomes experienced. Imagine a young child standing beneath a tree on a warm afternoon. Sunlight moves through leaves. Dust floats through the air. Pollen settles on clothing. Microbes live on the child's skin. Others inhabit the mouth and intestine. From the outside, nothing remarkable seems to be happening. The child is simply playing. Yet inside, barriers are working. Mucus is trapping particles. Macrophages are surveying tissues. Lymphocytes are circulating. Antibodies are binding. Cells are learning. Memory is forming. Ordinary childhood is an immunological education. Years pass. The thymus remains especially active during childhood. New T cells emerge. B cell populations develop. The immune system becomes more experienced with each season. Then adolescence arrives. Hormones change. The body grows. The thymus begins a gradual process called involution. It becomes smaller relative to the body, and more of its tissue is replaced by fat. This does not mean T-cell immunity suddenly disappears. Far from it. Large populations of mature T-cells already exist. Memory cells remain. New T-cells can still be produced, though thymic output generally declines with age. The system shifts. Adulthood brings another long period of immune experience. Every year adds new encounters. Some forgotten, some remembered, some leaving only faint biological traces. The immune system becomes a record of the environments through which a person has traveled. Two people of the same age may therefore possess very different immune histories. One may have grown up beside a farm, another in a dense city. One may have lived on several continents. Another may have remained in the same town. Their diets may differ. Their microbial exposures may differ. Their vaccinations may differ. Their infections differ. Their genes differ. Even identical twins can develop differences in immune function as their lives diverge. Immunity is biology meeting biography. And perhaps this is one of the most beautiful ways to think about your immune system. It is not simply something you were born with. It is something that has traveled with you. It was there on your first day of school, there through forgotten winters, there during every journey, every meal, every night of sleep, every cold you recovered from, every small cut that healed, every vaccination, every breath. Your immune system has been present for all of it. Now imagine walking through a long gallery. On the walls are no photographs. Instead, there are tiny glass cases. Inside each is a memory cell. One represents an encounter from childhood, another from adolescence, another from only months ago. Some are B cells, some are T cells, some may remain for years, others may eventually disappear. The gallery is constantly changing. New exhibits arrive, old ones fade, and you never consciously visit this place. It exists entirely within biology. As adulthood continues, the immune system also encounters something it must manage every single day: stress. Stress is not purely psychological. It is physiological. The nervous system and endocrine system communicate constantly with immune cells. Short-term stress responses can alter immune activity in ways that may be adaptive in particular circumstances. But prolonged stress can influence immune regulation differently. Hormones such as cortisol participate in these relationships. Cortisol is not a bad hormone. Your body needs it. It helps regulate metabolism. It follows a strong daily rhythm. It affects inflammation and immune function. The important idea is not that stress simply turns immunity on or off. The relationship is much more nuanced. Imagine the body preparing for a difficult day. The brain perceives a challenge. Signals travel through the nervous system. Hormones enter the bloodstream. Heart rate may change. Energy becomes available. Immune cells receive some of those signals too. The whole organism adjusts. The mind does not experience stress in isolation. Every emotional state occurs within a body. And the reverse is also true. Immune activity can influence how the mind feels. Inflammatory signals can contribute to fatigue. They can alter sleepiness. They can influence appetite and motivation. This is especially obvious during illness. But quieter versions of communication occur all the time. Brain and immunity are not two strangers exchanging occasional letters. They live in the same house. Which brings us back to sleep, perhaps the most appropriate place for our journey tonight. Sleep is not simply the absence of waking. It is an active physiological state. The brain changes its patterns of electrical activity. Hormone levels shift. Body temperature follows its nightly rhythm. Memory processes change. Metabolism changes. And immune function changes too. Researchers have found relationships between sleep and immune memory. Adequate sleep around the time of vaccination, for example, has been associated in some studies with stronger aspects of antibody responses compared with significant sleep restriction. Sleep also influences the movement and signaling of immune cells. Again, we should resist turning this into a simple rule. More sleep does not mean endlessly increasing immunity. The body prefers balance. But normal, sufficient sleep is part of healthy immune regulation. So tonight, sleep is not wasted time. Your conscious mind may disappear into dreams, but your physiology continues. The immune system keeps its own schedule. Imagine midnight inside a lymph node. There are no clocks on the walls, but cells respond to rhythms created throughout the body. Hormonal signals change. Patterns of cell movement shift. Lymphocytes enter and leave. Some remain close to antigen-presenting cells. Others travel onward. The night has a biological texture, different from noon, different from sunrise. Outside, darkness covers half the planet. Millions of people are asleep. Inside each of them, circadian clocks are influencing gene activity. Nearly every tissue has molecular machinery capable of keeping approximate daily time. The central clock in the brain helps coordinate these rhythms with the cycle of light and darkness. Immune cells participate in this temporal world too. The immune system does not simply know where it is. In a biochemical sense, it also responds to when. Imagine one macrophage in the skin. Morning changes the hormonal environment. Then afternoon, then evening, then night. The cell's activity can be influenced by rhythmic signals and local conditions. Tomorrow, the cycle repeats. Again and again, you have lived through thousands of these cycles. Your immune system has traveled through every one. Now we move towards something else the immune system encounters every day. Food, not as calories, not as flavor, but as foreign biology. A strawberry contains proteins your body did not make. So does rice, bread, fish, beans, milk. Nearly everything we eat contains molecules originating from other organisms. And yet a healthy immune system usually does not attack food aggressively. Why? Because tolerance is actively maintained. The digestive tract has evolved to exist in constant contact with food antigens. Specialized immune pathways help prevent unnecessary inflammatory responses to ordinary dietary molecules. Regulatory T cells participate. Intestinal epithelial cells participate. Dendritic cells can promote different types of responses depending on context. The microbiota contributes to the environment. The liver also helps maintain tolerance to many molecules arriving from the intestine. Eating is therefore an act of remarkable immunological trust. Imagine dinner entering the stomach. Proteins begin breaking apart. Acid unfolds them. Enzymes cut them into smaller pieces. Farther along, additional enzymes continue digestion. Eventually, amino acids and small peptides are absorbed. Carbohydrates become simpler sugars. Fats are processed and absorbed through specialized pathways. The meal gradually stops being something separate from you. Its molecules enter your body. Some will become energy. Some may become muscle. Some become enzymes. Some become hormones. Some may eventually become antibodies. A molecule that once belonged to a plant growing beneath sunlight may one day become part of an immune protein circulating through your blood. The boundary between world and self is always moving. This makes the immune system's task even more extraordinary. What counts as you? At the atomic level, The material of your body is constantly changing. Water enters and leaves. Carbon moves through metabolism. Minerals are exchanged. Cells die. New cells appear. Yet immune identity remains sufficiently stable for the system to recognize the body as self. You are a pattern maintained through changing material. Like a river, the water is always moving. The river remains. Now imagine that river in moonlight. Its surface reflects stars. Water passes beneath you. No drop remains forever. Yet the shape of the river continues across years. Your body is similar. Matter flows through. The organization persists. The immune system helps protect that organization. And sometimes, the distinction between self and other becomes beautifully complicated. Pregnancy is one of the clearest examples. A developing fetus carries genetic material from both parents. From the mother's immune perspective, the fetus is not genetically identical to her own tissues. Yet successful pregnancy usually proceeds without the immune system simply rejecting the fetus as it might an incompatible transplant. How? Through an extraordinary network of local immune regulation. The placenta plays a central role. Cells at the maternal-fetal interface display unusual patterns of immune-related molecules. Regulatory immune populations contribute. Local signaling. Pregnant people still need protection against infection. Instead, immunity is carefully adjusted. Tolerance and defense coexist. Another balancing act. Imagine the placenta as a peaceful border between two closely connected biological worlds. Nutrients cross. Oxygen crosses. Waste products move in the opposite direction. Antibodies can cross. Hormonal signals move. Yet the boundary remains. Two circulations come extremely close without normally mixing directly. And around that border, immune cells maintain a delicate conversation. Months later, birth occurs. The cycle begins again. Generations continued. Imagine a human family tens of thousands of years ago. Night has fallen. A small fire glows beside a shelter. Beyond the light is darkness. People sleep close together. There are no antibiotics, no vaccines, no understanding of microbes. Yet their immune systems contain the same fundamental families of cells we have been exploring tonight. Neutrophils patrol their blood. Macrophages live in their tissues. T cells circulate. B cells make antibodies. Bone marrow produces new cells. Lymph nodes organize responses. The biology is ancient. Those early humans knew illness, of course. They knew wounds. They knew recovery. They learned practical ways of caring for one another long before they understood the mechanisms. Rest, warmth, water, food when possible, cleaning wounds in whatever ways their cultures discovered. Human beings survived not because immunity was perfect, but because immunity, behavior, social care, and adaptation work together often enough. Our ancestors passed their genes onward, generation after generation. The immune system you carry tonight contains the legacy of survivors, not a flawless system. Evolution never produces perfection, but a system flexible enough to help human beings live in forests, deserts, mountains, coasts, grasslands, and eventually enormous cities. Cities created new immune challenges. When humans began living in dense settlements, Infectious diseases could spread through populations in ways that were difficult in small, scattered groups. Agriculture brought humans into closer contact with domesticated animals. Trade connected distant populations. Ships carried people, goods, rats, insects, and microbes across oceans. History became, in part, a history of immune systems meeting unfamiliar pathogens. Yet we need not turn tonight's journey toward frightening epidemics. Instead, consider the quieter consequence. Every generation learned, not consciously at first. Biologically, immune memory formed within individuals. Genetic variation influenced populations over longer periods. Human behavior changed. Eventually, science appeared. Sanitation improved. Clean water systems expanded. Vaccination developed. Antibiotics transformed the treatment of bacterial infections. Public health began interrupting transmission before immune systems ever needed to respond. And this is worth remembering. Your immune system does extraordinary work, but it does not protect you alone. Modern human health is also protected by things outside the body. Clean drinking water, sewage systems, food safety, vaccination programs, hand washing, ventilation, medical care, antimicrobial medicines when appropriate, safe childbirth practices, public health surveillance, scientific research. Protection exists in layers outside the body just as it does inside. Imagine a glass of clean water beside your bed. It looks simple, transparent, still, yet behind that glass may be reservoirs, filtration, testing, pipes, engineering, chemistry, public infrastructure, and generations of scientific knowledge. The immune system benefits from all of it. Sometimes the best infection is the one the immune system never needs to encounter. This is another peaceful form of protection. Prevention, a clean surface, a safe meal, a vaccine, a washed hand, a mosquito net in regions where mosquito-borne disease is common, a well-designed sewer. Simple things can alter the course of human health. And yet, even in the cleanest modern environment, microbes remain everywhere. They should. A sterile planet would not be Earth. Microorganisms support ecosystems. They cycle nutrients. They help plants grow. They live in oceans and soil. They inhabit our bodies. Some produce foods we enjoy. Yeast gives bread its rise. Microbes transform milk into yogurt and cheese. Others participate in fermentation. Microbial life is woven into civilization. The immune system evolved not to separate you completely from this microbial world, but to help you live within it. That difference matters. You are not a fortress sealed against nature. You are part of nature. Imagine walking barefoot through grass in the early morning. Dew touches your feet. Soil lies beneath the roots. Microbes fill that soil. Fungal threads stretch through it. Bacteria surround plant roots. Tiny organisms move through drops of water. Above you, pollen drifts through sunlight. Microbial particles float through the air. Your skin touches the world. Your lungs breathe it. Your digestive system receives pieces of it. And still, your body maintains itself. The immune system allows openness. That may be its deepest achievement. A completely sealed organism could never eat, never breathe, never reproduce, never interact. Life requires exchange. Exchange creates vulnerability. Immunity makes exchange possible. Your lungs must remain thin enough for oxygen to cross. Your intestine must remain permeable enough to absorb nutrients. Your skin must allow sweat to escape. Your reproductive system must interact with another human being. Every opening creates opportunity and risk. So evolution did not close the doors. It placed guardians beside them. Mucus, cilia, antibodies, macrophages, T cells, B cells, complement, chemical barriers, microbial communities, layer upon layer, not walls against life, gatekeepers allowing life to continue. Now the night grows very soft. Perhaps you have already drifted in and out of sleep. Perhaps parts of the story have disappeared. That is perfectly fine. You are not studying. There will be no test in the morning. You do not need to remember anything. Let the words pass through like water. Imagine your immune system now as an ancient garden. There are stone walls around the edges. Not too high. Gates remain open. Streams run through the garden. Small bridges cross them. Trees grow beside quiet paths. Some trees are old. Others were planted recently. Gardeners move slowly between them. They remove dead branches. They watch for damage. They tend the soil. They do not uproot every unfamiliar plant. They observe first. Some newcomers are harmless. Some are useful. Only when something threatens the balance do the gardeners intervene. In one corner stands a library. Inside are records of old seasons. A winter when frost arrived early. A summer when insects damaged the leaves. A year when the river rose. The gardeners remember. So when familiar signs return, they respond more quickly. This is immune memory. Farther away is a nursery. New gardeners are being trained. This is the bone marrow. Some will travel through the garden only briefly. Others may remain for years. Some will never encounter the problem they were prepared to solve. Still, they are needed. A garden survives partly because it is prepared for possibilities. At the center is a quiet schoolhouse. The thymus. Most of its busiest years were long ago if the garden belongs to an adult. But generations of T cells once passed through its rooms. They learned one of the garden's oldest rules. Protect what belongs here, but do not destroy the garden while trying to save it. Beyond the schoolhouse, streams converge at small stations. Lymph nodes. Information arrives from distant parts of the garden. Cells meet. Signals are exchanged. Decisions are made. Then messengers depart. And above everything is the night sky. Dark, deep, patient. The garden has survived countless nights before this one. It knows darkness. It knows rest. It knows how to continue without your attention. So walk slowly toward a small house at the edge of the garden. A lamp burns inside. Open the door. The room is warm. There is a bed beside an open window. Outside, the leaves move gently. You can hear water somewhere in the distance. Lie down. The gardeners will continue their work. You do not need to help them. Somewhere, a neutrophil is moving through blood. Somewhere, a macrophage is clearing debris. Somewhere, a B cell is waiting. Somewhere, a T cell remembers. Somewhere, an old red blood cell is being recycled. Somewhere, intestinal microbes are turning fiber into useful compounds. Somewhere, cilia are moving mucus. Somewhere, bone marrow is producing tomorrow's cells. And somewhere, a molecular signal appears, is noticed, does its work, and fades. The body does not need every signal forever. It knows how to let things go. Perhaps you can do the same with the thoughts of today. Whatever happened this morning can become softer. Whatever remains unfinished can wait. Whatever you were planning for tomorrow does not need to be solved in this moment. Your body has entered the night. Let your mind follow. The garden becomes dimmer. The paths disappear. The stream becomes only a sound. The library closes. The schoolhouse grows dark. The gardeners become distant silhouettes beneath the trees, and the warm light beside your bed becomes softer and softer until even that disappears. There is only breathing now, a gentle breath in, a gentle breath out, and another, slower than before. The immune system continues, quietly, faithfully, without applause, without consciousness, without asking anything from you. And as you drift farther into sleep, perhaps one final thought can travel with you. Your body is not defended by one powerful guardian. It is cared for by countless small acts of cooperation. A barrier holding, a cell noticing, a molecule binding, a message traveling, a memory remaining, a wound closing, a response ending. Small things working together through every hour of every day and through every peaceful hour of the night. Outside, the world continues beneath the stars. Inside, the ancient garden remains quiet. The gates are watched. The rivers are flowing. The lights are low, and you are free to sleep